Universidad de Santiago se complace en dar la más cordial bienvenida a todos y todas. Gracias por el interés en acompañarnos la mañana de hoy en el evento científico virtual Neurosur 2021. Hoy daremos inicio al simposio científico Memoria desde la molécula a la conducta. Vamos a dar inicio a la actividad. A continuación, las palabras del Dr. Juan Manuel Solés y Cid, rector de la Universidad de Santiago.
Señoras y señores, antes de dar inicio a mis palabras, quisiera saludar al doctor Claudio Hetz, director del Instituto de Neurociencia Biomédica, BNI, al señor Eduardo Rivera, Managing Director, MIT International Science and Technology Initiative, MISTI, doctora Leonora Mendoza, decana de la Facultad de Química y Biología, doctor Marcelo Cortés, director del Departamento de Biología. Además, quisiera saludar a los destacados panelistas nacionales e internacionales, académicos e investigadores y estudiantes que se encuentran presentes. Junto con saludar a todas y todos los presentes, quisiera agradecer su participación en Neurosur 2021, que se enmarca en la iniciativa MISTI-USACH del Instituto Tecnológico de Massachusetts, MIT, y que además cuenta con el apoyo del Centro de Estudios Latinoamericanos David Rockefeller Derklas de la Universidad de Harvard, así como también el Instituto Milenio de Neurociencia Biomédica, BNI, y la colaboración a nivel nacional de la Universidad de Chile. Este simposio científico surge como instancia para actualizar y difundir las herramientas de última generación para el estudio del cerebro. Esta colaboración surge del interés de investigadores de la Universidad de Santiago de Chile, la Universidad de Chile, la Escuela de Medicina de Harvard y el Instituto Pickower del MIT, con el propósito de informar a la comunidad sobre los últimos hallazgos en el estudio de las funciones cerebrales. Como Universidad de Santiago de Chile, nuestro propósito es crear, preservar, difundir y aplicar el conocimiento para el bienestar de la sociedad a través de la docencia, investigación y vinculación con el medio. Para ello requerimos generar las condiciones, capacidades y oportunidades que posibiliten a las generaciones futuras hacerse cargo del avance del conocimiento, su transferencia y retroalimentación para el crecimiento y desarrollo del país en una sociedad global. Y es justamente eso lo que desarrollamos en esta actividad, donde reunimos a connotados investigadores quienes presentan los avances de sus trabajos. Iniciativas de este tipo fortalecen el trabajo científico para todas las ramas de las neurociencias, y en este caso particular, con énfasis en los mecanismos que subyacen la memoria, la toma de decisiones y cómo se ven afectados por diversas patologías como el estrés, Alzheimer, anestésicos, entre otros. Investigaciones que aportarán sustantivamente a un mayor conocimiento del cerebro y cómo el ser humano se va adaptando a las diversas circunstancias, tales como las vividas en el último año a raíz de la pandemia. Estoy convencido que esta iniciativa tendrá un impacto positivo en el avance de la investigación en neurociencias, así como en la apropiación del conocimiento por parte de estudiantes de pregrado, posgrado y académicos de áreas afines. Quedan todas y todos invitados al simposio y workshop que se desarrollarán durante los próximos días, y mucho éxito para los participantes. Muchas gracias.
Muchas gracias al rector Dr. Solesi por sus valiosas palabras, y ahora nos permitimos otorgar la palabra al director del Brain Neuroscience Institute, BNI, Dr. Claudio Hetz. Adelante, por favor.
Muchas gracias por la introducción. Estamos muy contentos de realizar este evento en estas condiciones tan particulares que nos han tocado. Entonces, Neurosur es la iniciativa científica del sur. Nace el año 2012 con el liderazgo de Andrés Kuff, nuestro actual ministro de Ciencia, como una instancia liderada por este centro para potenciar la calidad de la investigación científica y sus bases tecnológicas mediante alianzas entre institutos y centros de investigación de neurociencia con un foco latinoamericano. Neurosur aspira a mejorar la visibilidad de la neurociencia regional y a posicionarse como un faro científico que fomente la colaboración en el Cono Sur. En las primeras reuniones de esta iniciativa visitamos múltiples centros a lo largo de Latinoamérica, por ejemplo en Argentina, en Uruguay, en Brasil. Luego invitamos científicos a visitar nuestros centros acá en Chile, lo que nos permitió identificar temáticas de interés común para crear instancias de colaboración, sin tener grandes financiamientos, sino básicamente asociarnos entre nosotros por un interés genuino de hacer ciencia conjunta. En este contexto se realizaron pasantías de entrenamiento para investigadores y estudiantes de posgrado con el fin de formar capital humano de alto nivel, nuevas tecnologías y potenciar la competitividad científica de la región, además de levantar financiamientos locales y latinoamericanos para la investigación conjunta. Y esto de hecho lo logramos, y logramos iniciar proyectos cofinanciados, por ejemplo, entre Brasil y Chile. También levantamos proyectos latinoamericanos entre por lo menos 5 países en el área de la innovación. Y desde el año 2016 se incorporaron a Neurosur nuevos elementos, como la realización de un simposio internacional con expositores de primer nivel en el área de la neurociencia y un curso de especialización para estudiantes latinoamericanos. Estas instancias dieron la oportunidad a estudiantes chilenos y latinoamericanos de compartir con destacados investigadores también de otros países. Esto fue súper importante. Sin duda, Neurosur ha crecido con el pasar de los años y esperamos que año a año se incorporen nuevas instituciones a esta red, como lo estamos viendo en este nuevo evento. Hoy día, en su versión número 8, Neurosur fue organizada por los doctores José Ignacio Egaña y Antonello Pena, de la Universidad de Chile, por Francisco Flores, del Medical General Hospital de Harvard, en Boston, y Elías Leiva, de la Universidad de Santiago. Personalmente, quiero agradecer todo el compromiso y motivación que han puesto para llevar este evento a cabo, sobre todo con las dificultades que conlleva realizar un evento virtual durante esta pandemia. Es bastante complejo. Este año, además del simposio internacional, que abarcará temas de memoria y aprendizaje, desarrollaremos una tercera versión del curso Herramientas y Análisis en Señales Electrofisiológicas, en el cual participarán 25 estudiantes de Latinoamérica. Así que esperamos que disfruten de las actividades durante estas 2 semanas, en las que compartiremos importantes estudios sobre memoria y aprendizaje, con una mirada desde la molécula hasta el comportamiento. Así que quería darles las gracias por su participación y poner todas las energías en preparar este evento, que va a durar 2 semanas. Muchas gracias.
Agradecemos la valiosa intervención del doctor Claudio Hetz y damos la palabra al representante del MIT, el señor Eduardo Rivera. Adelante, por favor.
Muchas gracias, Elías. Buenos días. Primero me gustaría agradecer a todas las organizadoras por la invitación para estar aquí hoy. Como director del Programa de Iniciativas Internacionales de Ciencia y Tecnología del MIT, MISTI, para Chile y otros países de la región, me enorgullece dar la bienvenida a todos los científicos que estarán presentando en los próximos días y a todos los asistentes a la conferencia Neurosur 2021. Como mencionaba hace algunos minutos el señor rector, algunos de los investigadores de esta conferencia son parte del Fondo Semilla de Investigación entre la Universidad de Santiago de Chile y MIT. En el pasado, también hemos tenido la oportunidad de colaborar con algunas otras entidades que patrocinan y organizan este evento, como el Instituto Milenio de Neurociencia Biomédica. Hemos tenido la oportunidad de tener algunos pasantes del MIT trabajando en este instituto de alto nivel. Hemos también tenido la oportunidad de colaborar con la Facultad de Medicina de la Universidad de Chile y en algunas actividades en conjunta en el pasado con el Centro Rockefeller para Latinoamérica de la Universidad de Harvard. Desde el 2011, el programa que lidero, MISTI, ha sido el puente para facilitar la colaboración de más de 150 científicos de universidades chilenas con un número similar de científicos de MIT. Estas colaboraciones iniciales, bajo el paraguas de un acuerdo de Fondo Semilla, han resultado en acceso a financiamiento adicional, a publicaciones, patentes y a conferencias. Un resultado igualmente importante y que fue parte de la motivación inicial cuando comenzamos a trabajar con la Universidad de Santiago, pero que sin duda mucho más difícil de registrar y hacer un seguimiento, es que los investigadores de universidades chilenas y de los Estados Unidos han ampliado sus redes científicas y han continuado colaboraciones más allá de los aspectos formales. Estas colaboraciones no habrían sucedido sin MISTI. Estoy seguro que el señor rector Solesi está muy complacido de ver que la colaboración que buscaba fomentar entre la Universidad de Santiago y la red científica asociada a la universidad y MIT se hace concreta con estos eventos, como el simposio que estamos dando inicio hoy. La ciencia ha sido históricamente una actividad que traspasa límites nacionales. En un artículo de 2015, Wagner y otros señalaron que la colaboración global, medida como autorías de artículos publicados en revistas con peer review y entendida como un subconjunto del total de la cooperación científica, continúa creciendo. El porcentaje de los artículos con autores internacionales se ha más que duplicado en los 20 años anteriores y representa todo el crecimiento de la producción de los países científicamente avanzados. Por supuesto, en estos tiempos sería imposible no mencionar COVID. Las colaboraciones internacionales han sido una parte fundamental de la lucha contra la pandemia, poniendo nuevamente en relieve el valor de las alianzas científicas globales. Desde el segundo fin de semana de enero de 2020, cuando se publicó la secuencia completa del genoma del coronavirus, hasta 11 meses después, cuando la primera vacuna, desarrollada también por un consorcio internacional, recibió autorización de uso de emergencia en los Estados Unidos, se han registrado niveles de colaboración científica internacional sin precedentes. Según la versión más reciente del Science, Technology and Innovation Outlook de la OCDE, entre enero y noviembre de 2020 se publicaron alrededor de 75.000 artículos científicos sobre COVID. Estados Unidos y China fueron los principales contribuyentes y una cuarta parte de los artículos se realizó en coautoría con investigadores de otros países. A pesar de la agitación política y económica, el más alto nivel de colaboración fue entre científicos de Estados Unidos y China. Al mismo tiempo, 75% de los artículos científicos sobre COVID se publicaron con acceso directo. Las editoriales científicas eliminaron los muros de pago en torno a la investigación de COVID, lo que hizo que el contenido estuviera disponible gratuitamente para que todos los investigadores pudieran acceder. La respuesta de la ciencia a la pandemia nos proporciona una potente ilustración del valor de la colaboración global y su impacto. Esta vez se ha dado en un contexto con un nuevo invitado sentado en primera fila, el público general. Más allá de las actividades de investigación, se ha requerido de los científicos que proporcionen información experta sobre ciencia, salud pública, e incluso se les requiere sobre las respuestas políticas a la pandemia. Muchos de los nuevos comunicadores científicos se han convertido en personalidades de medios de comunicación, intentando comunicar basados en una evidencia que inevitablemente cambia y necesita ser complementada. Es de esperar que el interés público por la ciencia se mantenga aun cuando la pandemia esté superada. A pesar de las interrupciones, limitaciones, desafíos de movilidad física y un sinnúmero de obstáculos, los científicos de todo el mundo han continuado su trabajo durante la crisis, utilizando herramientas digitales, infraestructuras de datos abiertos, encontrando formas creativas y nuevas de compartir, colaborar y continuar funcionando fuera de sus entornos habituales de laboratorio o de campo. Este seminario es una clara evidencia de aquello. Para muchos, la crisis del COVID ha acelerado las tendencias que ya estaban en marcha. Ha abierto aún más el acceso a datos y publicaciones, ha aumentado el uso de herramientas digitales, mejorado la colaboración internacional, estimulado una variedad de asociaciones y alentado la participación activa de nuevos actores. Es de esperar que estos desarrollos sean sostenibles y que acelere la transición a un panorama internacional de ciencia e innovación aún más abierto y colaborativo. Sin más que agregar, y dándoles las gracias, solo me queda desearles a todos un simposio productivo y, por qué no decirlo, sin problemas de conectividad de Zoom. Gracias.
Muchas gracias a Eduardo Rivera. Ahora agradecemos además la presencia de nuestra decana de la Facultad de Química y Biología, doctora Leonora Mendoza, del director del Departamento de Biología, doctor Marcelo Cortés, y de la directora de Relaciones Internacionales, Anouk Vandenberg. Gracias por acompañarnos el día de hoy. Ahora, sin más preámbulo, damos inicio a la actividad. Primero les recordamos que este simposio científico, Memoria desde la molécula a la conducta, está estructurado en 4 sesiones que se realizarán desde hoy, 5 al 8 de abril del 2021. Todas las sesiones se realizarán a través de Zoom y todos los participantes interesados recibirán los enlaces de cada sesión día por día. La primera sesión de hoy estará enfocada en los mecanismos de coordinación neural y estará moderada por el doctor Francisco Flores del Massachusetts General Hospital and Harvard Medical School. Las preguntas deben ser realizadas a través de la opción de preguntas y respuestas ubicadas en el panel que está bajo de sus pantallas. Y ahora le damos la palabra a nuestro moderador, doctor Francisco Flores. Muchas gracias y bienvenidos a todos.
Muchas gracias, doctor Leve. Muchas gracias a todos los que introdujeron este simposio. I'm going to switch to English for the rest of the session. So as Elias already said, I want to remind everyone that there will be 5 to 10 minutes at the end of each talk to do questions, which should be done through the option that you have in the bottoms of your screens. There will be a 15-minute break after the first 2 talks at around 10:45 a.m. and the session ends at 12:30 p.m. So without further ado, welcome to the first session of the symposium. This session is dedicated to mechanisms of neural coordination, and we will review several features of anatomical and functional brain coordination and how their normal and pathological function affect memory and learning. And I have the honor to introduce our first speaker of the day, who is Dr. Matt Wilson. Dr. Wilson is the Sherman Fairchild Professor of Neurobiology in the Picower Institute for Learning and Memory, the Department of Brain and Cognitive Science, and the Department of Biology, all of them at the Massachusetts Institute of Technology. Dr. Wilson's work focuses on the neural mechanisms of memory consolidation by studying interaction between the hippocampus and other brain structures during spatial navigation and sleep. Dr. Wilson is also responsible for the development and refinement of the multi-electrode recording technologies in freely moving animals, and it is behind the development of the OpenFIS system for in vivo electrophysiology. Dr. Wilson, welcome, and thank you so much for being with us today.
Great, thank you, Francisco. And thank you to all the organizers. Real pleasure to be here. It's a great event. I always enjoy participating. I'm just gonna go ahead and share my screen. Hopefully that worked out. So as Francisco said, my talk, I'm gonna review a lot of the work that we've done trying to understand both the mechanisms and function of hippocampal memory processing and the role of sleep. I'm just going to start off here showing you an illustration of the hippocampus in the rodent, where you see this cutaway view, the classic view of the hippocampus and its trisynaptic loop, where information comes from across the brain, converges in the entorhinal cortex, and then passes through this series of synaptic connections in regions within the hippocampus, the dentate gyrus, CA3, CA1, Zopiclon, then back out again. And some basic observations about the hippocampus, the discovery of its role in memory with the seminal case of the patient HM, who underwent a surgical procedure involving bilateral resection of the hippocampus and subsequently lost the ability to form any new memories of experience or events in his life. And then the discovery in the early 1970s by John O'Keefe by recording from individual cells in the hippocampus, individual neurons in the hippocampus, that these neurons had spatial receptive fields, that they responded to where the animal was in space. And this gave rise to what was described as the cognitive map hypothesis, that the hippocampus forms an internal map of space that's used both in navigation and forming memory. So those two observations, that is in humans, hippocampal involvement in episodic or event memory, and in rodents, the expression of spatial receptive fields, or place fields as they were referred to, and the role of the hippocampus in navigation, raised the question of what the common function of the hippocampus in these sort of two organisms is, the human and the rodent. Hippocampal structure is largely evolutionarily conserved, and so it's the same structure performing seemingly two different roles, but you can think of these roles as having one common element, and that is the need to encode temporal sequence information. That is to capture in space, spatial navigation, encoding the sequence of spatial locations, and episodic memory, the sequence of events. And of course, forming memory or understanding the temporal ordering of events in the world is essential for planning, for understanding causal relationships. Did A cause B? For directing actions, taking an action, leading to an anticipated outcome. So clearly, being able to form memories that capture temporal sequence information and then using temporal sequence information to guide behavior and future memory formation is something that's critical. So to study this basic function of the hippocampus, memory encoding, space, and the potential role of temporal order, temporal sequence information, as Francisco mentioned, We developed techniques for recording from collections of individual neurons using these wire electrodes, described here as a tetrode, which is a very small bundle of wires. This small bundle of wires is about 30 microns in overall diameter. The electrodes are placed into the brain adjacent to individual populations of individual neurons, and the discharge or action potentials produced by these neurons is then recorded. And this is a device that a microelectrode microdrive array, which allows the implantation of multiple microwire electrodes. And this shows some actual data, a cartoon here illustrating the multi-wire electrode placed next to, let's see, 2 cartoon neurons here, but in the actual brain, the discharge of those neurons produces action potentials where the amplitude of those action potentials varies depending on the distance. So a neuron that's close to, for instance, this wire will produce an action potential as a large amplitude. If it's farther away from this wire, there'll be a smaller amplitude. So you can use this relative amplitude and displaying individual action potential events collected over about 10 minutes here. You can see that they form clusters based on the relative position or distance of these neurons from the wire. So single electrode can record multiple neurons. When you take these neurons, the clusters that we identify as potential neurons, and then display their activity over about 10 minutes as an animal explores in space, you see illustration here of the phenomenon first discovered by John O'Keefe, and that is the place cell or place fields, in which individual colored points here are single action potentials, and then they're shown on the location on the track where the animal was when the colored neurons discharge. So the yellow neuron fires whenever the animal is running along this track in this direction. Is a top-down view of about a 2 meter track. And when the animal runs here, the yellow cell fires, the red cell fires here, the green cell fires here. So this sort of illustrates both the spatial encoding and also suggests that because of the spatial receptive fields, when an animal runs through these fields in a circumscribed trajectory, that is running along this path, the cells will fire in order: yellow, red, green. When the animal turns around and runs in the other direction, these cells also fire, not just as a function of position, but also direction. So in this direction, the blue cell fires, and there will be other cells that will fire. So there are different sequences of spatial firing in the 2 directions. If you place the animal on another track in the same space, you get a different collection of cells, different place fields, different spatial sequences. So this is the basic mechanism of spatial and spatial sequence encoding, but there's another element to hippocampal activity and hippocampal encoding, which is not captured by just looking at the total activity or the spatial bias over 10 minutes. There's actually a dynamic component, and that is a temporal component. So in time, activity in the hippocampus is modulated by at least 2 fundamental rhythms. During active behavior, a 4 to 12 hertz rhythm known as the theta rhythm, which you can see here illustrated in some early work by Yuri Busaki, in which he's recording the local field potential or the electrical activity produced by populations of cells. And you can see that during behavior, this electrical activity oscillates in the range of sort of 4 to 12 hertz. When the animal stops very quickly, within about half a second to a second, that activity transitions into these irregular spikes of activity, and these sharp waves. And on top of these sharp waves, you can't see it here because of the temporal resolution, there are high frequency oscillations known as ripples. So 2 fundamental modes, a theta rhythmic mode, and then during active behavior, and a sharp wave ripple mode during immobility, or as we'll see also in sleep. So we can focus on this theta rhythm. So when the animal is moving, you have both firing as a function of location, but also firing as a function of this rhythm. And I think it's easy to illustrate that here in some raw data. If you listen, you hear the spiking activity and also see the color-coded firing of cells. So for instance, the light blue cell fires here, the darker blue cell will fire here. And if you listen carefully, you can hear this rhythmic kind of rhythmic modulation of activities in animal moves. As soon as the animal stops, the rhythm goes away, and you hear these bursts, or kind of here the animal stops. Wait. There's a burst of activity. That's sharp wave ripples. When the animal moves again, the rhythm will return. So here you'll hear this. It might be harder for some people to hear that, but if you listen to a lot of brain activity, these rhythms really jump right out. And so you can, for instance, right there, moving, rhythm, stopping, sharp wave ripple burst. Now we can take that same activity, but instead of just illustrating the raw spiking activity as a function of position, we're going to use this strategy of ensemble decoding. And that is, we're going to take the activity of a collection of neurons at any given time and estimate the location of the animal that would give rise to that pattern of activity. And that is, if we know where the animal is, we can say which cells are likely to fire. For instance, if the animal is here, you could say it's a high probability that the dark blue cell will be firing here. Now you can invert that. If you hear the dark blue cell firing, you can estimate what's the likelihood that the animal would be in any given location. You can say, well, the probability is high here and it's low everywhere else. And so that's for a single cell. We can do this across a population and estimate the likely position of the animal based on ongoing pattern spiking activity in the population. And that probability or likelihood is going to be shown as a triangle where the size of the triangle is the magnitude of the probability. And it's also pointed in the direction that we estimate the animal's moving in, given that pattern. And so here I'll just show you this movie. So then this is decoded activity. And so now we're every 200 milliseconds, we're estimating what's the likely location of the animal. The green circle shows where the animal actually is. The blue triangles show where the decoded position, reading the brain of, or reading the mind or the hippocampus of the animal, tells us where the animal is. Now you notice when the animal's moving, the blue triangles correspond pretty well to where the animal actually is. That's the spatial coding. When the animal stops and you get those bursts, you notice the triangles jumped all over the track. So in these two states, during active mobility, movement, hippocampus seems to code for the animal's current location. During immobility, the hippocampus expresses a code that corresponds to remote locations. And so we're going to take a look at that coding initially of location during active behavior first. And if we zoom in now, we're going to do the same thing now, decoding, but now at a much finer time scale. So instead of every 200 milliseconds, we're going to be decoding every 20 milliseconds. That allows us to resolve or decode within individual theta cycles. So here is an example of a couple of seconds of theta modulated activity. You can see the local field potential. This is the theta oscillation. We're recording neural populations, and now we're going to decode every 20 milliseconds. And now you can see this is the actual position of the animal as it moves over those few seconds, the dotted line. And when we decode the position, you see that the decoded position, while on average actually follows the animal, in detail at higher resolution, the decoded position actually sweeps. It has these short sequences that go from a position just behind the animal to just in front of the animal. And it does this every 100, 120 milliseconds or so. So about 10 times a second, the actual hippocampal code is not just a positional code, but it's a repeated short sequence code. And going from just behind the animal, you could think of where the animal just came from, to a point, to a position in front of the animal, or where the animal is going to. So behind, in front, past, future, repeatedly played out 10 times a second, locked to the hippocampal theta rhythm. So that's the high temporal resolution code. Now I want you to sort of keep this in mind. That is a code, a sequential code that lasts about 100 milliseconds and captures, in this case, you can see the spatial scale, about 50 centimeters or so of space. And so that's during active movement, theta coding of these very short sequences. But what about when the animal stops? And so here's an example when the animal stops at this location. You remember you heard this kind of burst, this kind of sharp wave ripple activity. Well, it turns out this sharp wave ripple activity comes not just as a random burst of activity, but it comes as a burst of activity that lasts, in this case, about half a second. So there's a period of elevated multi-unit activity, lots of cells firing, and that firing is modulated by these high frequency oscillations, sharp wave ripples. We've taken out I kind of showed you on the Busaki diagram, there were these sharp spikes. We took out the lower frequency, the sharp spikes, so you can see the high frequency oscillations or ripples. So you can see when the animal stops over about half a second, there's an elevated probability of firing of the population, lots of cells firing, and that their firing is modulated by these high frequency ripple events, each of which lasts on the order of about 100 milliseconds or so. Now, when we decode that activity, sharp wave ripple activity bursts over about half a second, what we see is that just as when we decoded activity during behavior at a fine temporal resolution, if we do the same thing during the stationary period, we see that those the little triangles that seem to jump across the track actually form sequences, extended sequences that that go from, in this case, where the animal is currently to a location several meters away. This is a 10-meter track, so this is about 5 or 6 meters away. And interestingly, you could see that this extended sequence, this is time and this is position, seems to come in the form of a chain of shorter sequences. And that is a long sequence broken up into smaller chunks. There's 1, 2, 3, 4, or the smaller chunks of that sequence. And so I'll just show that here, decoded. This is slowed down so you can actually see it. So this is slower than real time. And so again, you can see the triangles, the decoded position going from where the animal is and following a path about 5 meters away. Now, there are other instances, for instance, the animal now has stopped here at another point where you see a sequence expressed, but interestingly, it can be going in the time reversed order. And so here, it's a sequence. I'll play that again. But notice the triangle is pointed to the right, but moving to the left. And that is the animal, as though the animal would be pointed in this direction, but moving backward, as it would be moving backward along the path. This illustrates a property of what we refer to as these replay events, where the sequences can be expressed in both time forward and time reversed fashion. So we have sequences that seem to be encoded during the theta oscillation, re-expressed during periods of quiet wakefulness in the form of sequences of shorter chunks or sequences. And when we look at those extended sequences of sequences, we see that the individual chunks correspond to individual sharp wave ripple events. So longer sequences lasting about half a second are actually comprised of a series of short 100 millisecond or so sharp wave ripple events. And again, the time scale of the sharp wave ripple is approximately the time scale of the theta sequence. So you can think of the fundamental unit of encoding as a theta sequence, 100 milliseconds in duration. In the case of this example, 40 to 50 centimeters in length. And then you chain those together to get longer sequences by chaining together multiple sharp wave ripple events. And this is during quiet wakefulness, but the same kind of activity can be observed when the animal's asleep. So here the animal has completed the behavioral testing session, been taken out and put into a separate box. Here you can see in the inset, the animal, this is just showing you an overview of the actual behavioral apparatus. The animal's not in that apparatus. It's been taken off. It's in a sleep box where you can see it's curled up and sleeping. And now we're recording activity during sleep. And what you see is, but decoding it on the track. And so you can see that we see similar decoded sequences. Just play that again. Here's a sequence that gets played back. It's playing back a sequence on the track. So during sleep, you get remote reactivation of these sequences on the track that can come both in the time forward, time reverse fashion, having a structure very similar to that of quiet wakefulness. Now, during sleep, one of the characteristics of sleep, unlike quiet wakefulness, is that sensory cortical areas go into a different state, and that is they're in this offline state as well. And that can include, in the case of the visual cortex, the animal sleeping in eyes closed posture, the absence of external sensory information. And the activation of sensory cortical areas during sleep in the absence of external stimulation, you could think of as being perhaps the basis for the subjective experience of dreaming. And that is that during sleep, you're imagining events, that there's some visualization that occurs, and we think of that as a dream. So one question is, do rats in this case show similar kind of sensory cortical activation? Do they dream in a sense? And we can do that by recording in both hippocampus and in and in visual cortex. And so here in an experiment by Daeun when he was a postdoc in the lab, having an animal run along a track similar to the one I showed you earlier, but this has visual patterned inserts along the track itself, different visual cues, different patterns at different locations. When an animal runs on this track, what you find is that individual visual cortical cells also show spatial receptive fields. They fire in different locations. And here's a series, animal walking along a path. Here's a series of 8 to 10 visual cortical cells. And you can see that these visual cortical cells also fire in a sequence, different visual cortical cells firing at different locations of the track. Here, a set of hippocampal place cells also firing in a sequence. So in this task, recording in the visual cortex and the hippocampus, you can get visual cortical sequences, hippocampal sequences. Just shown here, this is just the smooth average firing rate, just showing this is the average sequence of these cells on the track in the visual cortex, the average sequence in the hippocampus. During sleep, there are these periods lasting on the order of half a second to a second, where you see increased spiking activity and you see the expression of sequences in the visual cortex and in the hippocampus. Now, one interesting property of these sequences in the visual cortex and the hippocampus is if we zoom back out, so that was on a short time scale of about a second or so, but if we zoom out into the multi-second, 10-second time scale or so, what you see is that these half a second or so periods of elevated activity come in repeated fashion, and that is that this repeated organization corresponds to what we refer to as the slow oscillation during sleep. And that is sort of a repeated switching from periods of elevated activity to silence, to elevated activity and silence. If you were recording in individual cells, this transition to elevated activity is referred to as an up state. The period of silence is referred to as a down state. The up state corresponds to depolarization or increased excitability of neurons. The down state corresponds to hyperpolarization, decreased excitability. So in the visual cortex, in this case, large populations of cells are all being excited. There's lots of firing during the up state, and then they're silent. And then they fire, and then they're silent. And this repeated cycling between up state and down state is referred to as the slow oscillation. Now, in the hippocampus, you see a similar pattern of activity, periods of elevated activity, upstate like activity, and then downstate activity. Unlike the visual cortex or neocortex in general, cells in the hippocampus don't spontaneously actually switch between up and down states. If I took a culture or a slice of the neocortex, isolated these cells, the isolated cells are capable of switching into these depolarized and hyperpolarized states. The hippocampal cells don't have that property. So what we see here, this up and down state like activity must be driven or inherited from the neocortex. And you can see that also evidenced in the timing. And that is the up state, a transition to increased activity in the visual cortex precedes the transition to an upstate in the hippocampus. So visual cortex turns on, then shortly after that, within about 100 milliseconds, the hippocampus turns on, visual cortex turns off, and hippocampus turns on. So again here, visual cortex, hippocampus, visual cortex. So the visual cortex leading the hippocampus. But also you notice the visual cortex ends and the hippocampus ends. So you can actually see here that there's kind of a sequence of sequences. Visual cortex, hippocampus, visual cortex, hippocampus, kind of a back and forth. And so this suggests that this extended sequence of of upstates could actually correspond to a dialogue or a sequential dialogue between the hippocampus and the neocortex. Now, if it is the case that the neocortex drives the hippocampus and that there's reactivation of sequences in the visual cortex and the hippocampus, one question is, are the sequences or memory events, memory sequence of the hippocampus, driven by activity in the neocortex? Can the neocortex bias hippocampal memory sequences? And one way to test that is by taking advantage of a property of sensory neocortex during sleep. And so here, this just kind of illustrates this, the basic concept of neocortical upstates preceding hippocampal upstates, and the neocortex turns on. That then drives the hippocampus via the entorhinal cortex, and that is gated through the thalamus. And some of the later talks today are going to touch on some of these properties of both thalamic gating and hippocampal neocortical dialogue. I'm not going to go into that in any detail, but just the general concept that there's this neocortex to hippocampus, but then hippocampus back to neocortex, both at the up and down state level, but then potentially also at this individual sharp wave ripple level. So that there could be a fine time scale dialogue going on within an upstate, but also a dialogue going on across upstates. And this idea of chaining of sequences or sequences of sequences with neocortex initiating this, and that is driving the hippocampus, and then this back and forth propagating this sequential activity on its own, can be tested by, again, leveraging a property of the hippocampus and or the neocortex and the neocortical hippocampal dialogue, and that is that that sensory neocortex, and in particular auditory cortex, remains responsive during sleep. And that is that that auditory cortex, you still hear things when you're asleep, you don't necessarily wake up. And that is that you have auditory sensory vigilance without having necessarily auditory sensory arousal. So you hear things, you don't necessarily wake up to them. And so Dan Bender, when he was a postdoc in the lab, developed a task involving auditory spatial conditioning. Animal hears, he pokes his nose in the center of this linear track. He hears two sounds, the left sound, which is this downward frequency sweep, or pew, or an upward sweep, weep, pew. So we have pew, weep, two sweeps. One indicates go right, the other indicates go left. Very simple task. Animal repeats this, gets good at it, then the animal goes to sleep. And then every 10 seconds or so, we randomly play either the left sound or the right sound, and then ask what happens in the hippocampus following the delivery of those sounds in sleep. This just shows some of the basic data, the basic task. The animal's going back and forth. We're decoding the position while it performs the task. You see place cells firing here as the animal performs the task. Here are place fields. Some place fields are on the right, some place fields here on the left. And so the right place fields are going to be associated with the right sound, left with the left sound. This is during behavior. And then, this is now zoomed out, the animal's running the task, the animal goes to sleep. You can see this pattern of activity, which corresponds to like these kind of sharp wave ripple, slow oscillation driven events during sleep. If we kind of zoom into one of these things, here you see one of these sharp wave ripple burst events, or kind of putative upstates. And we're going to zoom into that. And so here you see, here's a kind of a pattern of hippocampal activity that we decode into a sequence onto the track, the left side or the right side. So during sleep, we play some sounds every 10 seconds or so. We decode the activity in the hippocampus that follows from that. We determine where that activity would have occurred when the animal's on the track. Are these place cells on the left side or the right side? And then we ask, is that activity, the place cell activity, biased in some way by the sounds? And the hypothesis is when we play the right sound, we're going to activate place cells on the right. When we play the left sound, we're going to activate place cells on the left. Red sound will activate the red cells here. Blue sounds will activate the blue sounds. And the basic result is that's what's happened. That's what happens. That when we activate the red sound, the left sound, we tend to activate place cells on the left side of the track. and we get the converse when we play the right sound. We activate play cells on the right side of the rack. This is all, again, during sleep. This is just the overall bias. Now, one interesting property of this is that that bias is not simply confined to the period of sensory stimulation. So it's not just when we play the sound. It lasts or continues to be expressed for many seconds, up to, again, on average, it's about every 10 seconds we're playing the sound. So you play the sounds, and then for 10 seconds after that, all the reactivated events tend to show this bias. So there's something that continues to persist, and that is a bias in the reactivation that persists after the initial sound cued bias. And this idea that the bias in memory reactivation in the hippocampus actually has some memory function was illustrated in some psychophysical experiments that were done by colleagues, including Jan Born, who did this experiment using not auditory but olfactory stimuli, and then Rudoy and Ken Paler's lab, who did a similar experiment using auditory cueing in a spatial matching task. And this is now in humans. So humans are performing a simple spatial matching task here. It's a card matching where they see illustrations here of a cat, and they have to remember where's the other cat. When they see it and they flip it over, not only do they see the cat cards and their position, but they're also given a sound to associate with it. They flip over the cat, they hear a meow. They flip over teapots, they hear a whistle. And then during sleep, they play either the cat sound or the teapot sound, and what they find is when they wake up, they perform better, and that is that there is sort of an improvement, fewer errors for the cues, for instance, the cat that was cued during sleep. So if you play the meow sound, you're better at finding the cat when you wake up, suggesting not only is there involvement of sleep and memory, but there is very specific cue content, specific memories can be processed, and that the specific memories that can be processed can be biased using simple auditory cues that can get into the brain and presumably affect memory through this neocortical to hippocampal dialogue. And so just in summary, the idea that you stimulate the cortex, that can bias what the hippocampus replays. The hippocampus then plays that memory back and influences the cortex. And it's this dialogue that is the basis for sleep and memory processing. Now, you can kind of think of this is just a kind of a simple cartoon illustrating the different kind of hippocampal neocortical interactions that can occur during these slow oscillations. And within the slow oscillations, as I pointed out, there's actually a finer structure. It's not just increased activity, but in the neocortex, you see these events known as sleep spindles. In the hippocampus, you see these multiple ripple bursts. And so the idea is that there are sequences of sequences, a dialogue which involves rapid back and forth interactions within upstates, but then also potentially interactions across upstates. And so you can think of all of this as corresponding to an extended sequence of kind of state sequence evaluation. You could also think of this as like a coupled hidden Markov model, a model in which the hippocampus is trying to predict what's going to happen next. The visual cortex also trying to figure out what's going to happen next, and they're talking to one another. So this idea of search being involved not only in memory, but also in general cognitive processing or learning is quite powerfully and elegantly illustrated in current AI work, in which, for instance, AlphaGo, an algorithm which beat the best human Go player, used a strategy of deep search combined with online experience and offline simulation. So AlphaGo basically played itself, simulated many games, searching through or sort of running through sequences of moves during offline periods or sleep. And in doing so, became the best Go player in the world. Now, this idea of search combined with the evaluation or valuation of different moves required not only do you simulate the states, but you also have to figure out, well, how well did that work out? And as you have to combine simulated state sequences with value information. And the idea of combining value information or using value information with spatial sequences is quite familiar in the domain of navigation, where one has to do both forward planning, and an example of some experimental work by David Foster after he left my lab, illustrates that these replay events during quiet wakefulness can correspond to future trajectories leading to reward. So in this case, the animal was randomly foraging. There was a hidden reward location. The animal had to plan or search out where is that likely reward. And the replay events tend to be biased toward the reward location. So sort of suggesting a planning role for replay events that was biased by future reward. You can also think about the reverse sequences solving another problem involving reward, and that's the temporal credit assignment problem. And that is sort of learning after the fact, here best illustrated in trying to solve a maze. You wander through the maze, you finally find the exit, and what you need to remember is not where is the exit, that's the reward location, but how did I get there? That is, you have to propagate a value back along the path or give credit in time backward along the path. And this backward temporal credit assignment problem could be solved by pairing a reward signal. For instance, here, this is a cartoon dopamine activity profile paired with a reverse hippocampal sequence, turning a single reward location signal into a graded reward position signal in which the gradient points to the location of the reward. So the animal runs along here, finds a reward at this location, propagates the reward information back in time. And so now the next time the animal comes here, it could just follow the gradient to get to the reward. And so this suggests pairing of reward, replay, a transformation of reward signaling from a discrete location backward along a gradient. And this is consistent with many forms of classical reinforcement learning, and behaviorally has been confirmed as being the likely mechanism by which animals learn through repeated experience to drive behavior to achieve or optimize expected reward outcomes. So it's like this is the cartoon of probably how things work, but is that how they actually work out? Now, to answer that question, we have to go and record. And so Stephen Gompertz, when he was in the lab, recorded in the hippocampus and in the VTA looking at dopamine neurons. And in a simple task, a spatial working memory task involving starting at one location, either on the left or the right side. Here's a choice trial. You run up the central arm, and the task is go to the same side that you started on. So if you started on the right side, go to the right side. The animal then turns around and comes back. This is what I referred to as the forced trial. Comes back and then is forced into one of these other locations. So the animal's running back and forth in this direction, animal's choosing. In this direction, we're forcing or we're choosing. And what you find is, if you look at dopamine neurons, you find that they have a couple of different profiles. Here are a couple examples of dopamine neurons. Here, dopamine neurons that fire close to the reward location. The nose poke is where the animal gets the reward. Some neurons fire on approach to reward. Other neurons fire at the reward. There are also non-dopamine neurons that don't seem to show those sorts of behavioral correlates. Also, if you look at the average activity, you find that dopamine neurons, indeed, when the animal actually running up here gets to the choice point, you see this kind of ramping activity. So this suggests that during behavior, indeed, you find activity seems to correspond to the kind of expected activity predicted by reinforcement learning. Now, if we look during quiet wakefulness, the animal sitting and stopping, where in the hippocampus, you find these periods, this is activity in the hippocampus, of elevated multi-unit activity or sharp wave ripple-driven activity. You also see, and so here's some sharp wave ripples, these upstate-like periods. And then in the VTA, you find that there are also periods where the dopamine neurons fire. Dopamine neurons tend to fire at a relatively low rate, but if you look at this repeatedly, and so this is sort of a post-stimulus time histogram locked to the occurrence of sharp wave ripple events. So whenever there's a sharp wave ripple event, you ask, what's the hippocampus doing? You see lots of activity around sharp wave ripples. You also see in the ventral tegmental area, VTA, there's also an increase in activity. So the VTA responds to sharp wave ripples. It knows that the hippocampus produced a sharp wave ripple. Now we can ask, okay, it knows there's a sharp wave ripple, but does it know what's in the sharp wave ripple? And as we could decode the memory content, the spatial content within one of these events, this is a single event, for instance, we decode the position. This sharp wave ripple burst seems to express a sequence that kind of goes along the path here, runs along here, an extended sequence. And then we can divide these events into events that we can decode, or this is so-called replay events. So here's a sharp wave ripple burst event, which we can decode into a sequence in the track. Here's another event that doesn't seem to decode. It's replaying something else. It's not replaying activity that we can decode on the track. We refer to that as a non-replay event. And if we divide up these events and ask for sharp wave ripple events that contain decodable content, and we look in the VTA, we find, again, dopamine neurons seem to care about these decodable replay events. Other events, they also have sharp wave ripples, and so you can see the hippocampus still locks to the non-replay content. The VTA does not. So the VTA knows both that there are sharp wave ripple events and what is being replayed, in this case, replaying sequences on the track. And we can break that down even further. We can ask, okay, so the VTA knows that it's replaying a sequence on the track. Does it care what the actual locations within the sequence are? And so now we're going to look at the decoding of hippocampal position and the timing of dopamine neurons relative to the decoded position on the track. And that is, when dopamine neurons fire, what's the location that the hippocampus is replaying? And what we find is that the location that is being replayed in general is the goal locations. So the hippocampus replays a sequence, the VTA reactivates, fires when the hippocampus reaches goal locations. And if we put the animal on a different track, we also see hippocampal replay events, we see VTA activity, but we see less bias toward goal locations when the animal is running on a simple track where it does not actually have to make decisions. So we see that the VTA knows both what the hippocampus is doing, what the content of replay events are, and that is, are they on the track or not. They also know the location of goals and tend to fire when the animal expresses goal locations. And they also know whether those goal locations are tied to decisions, whether there's an actual choice task involved. And so you say all of that, that's interesting, but this is all occurring while the animal's during quiet wakefulness on the task. When the animal goes to sleep, as I illustrated earlier, the hippocampus reactivates sequential memory, just as that look very similar to that during quiet wakefulness. So we can ask, does the VTA also care about hippocampal replay, goal locations, etc., during sleep? And so here's an example of a VTA neuron during sharp wave ripple events, replay sharp wave ripple events modulated by sharp wave ripples. During sleep, on the other hand, sharp wave ripple replay events are not actually correlated with an increase in VTA activity. In fact, there's a slight decrease in activity, and that is dopamine neurons do not fire to replayed sharp wave ripple events. So that's interesting. It suggests that reward information is paired with hippocampal replay during quiet wakefulness, but is not paired with reward information during sleep. Same replay events, different reward contingencies pairing with it. Now that's quite interesting. This is my final slide. Given some behavioral work that was done, very clever work done by my colleague Karim Ben-Shanain, who used this closed loop manipulation, recording hippocampal play cells, electrically stimulating in the medial forebrain bundle, which will activate dopamine neurons in the VTA. And he leveraged this task known as place preference learning, where an animal wandering around in an arena, if it goes to a certain location and you stimulate dopamine release by stimulating the medial forebrain bundle, animals will very quickly learn to prefer that location. They like locations where they get reward or dopamine. What Ben-Shanain did was, had the animal wander around, did not stimulate it, but simply recorded place cells. And then during sleep, whenever a place cell, a particular place cell, in this case, for instance, the place cell that fired in this location, whenever that place cell fired, it would electrically stimulate. So he's stimulating dopamine release paired to place cell activity. And then the animal wakes up, puts him back in the arena, and the animal expresses a place preference. It likes the place that was paired with reward during sleep. This is completely de novo learning. The animal never experienced reward paired to this association during wakefulness. It was only during sleep. So it says that the hippocampus and the system is very capable of learning new things new reward associations during sleep, but for some reason, natural sleep tends to avoid that. So it turns off dopamine, at least during some sleep processing, precisely to avoid this kind of de novo learning. So what this suggests is that overall, the hippocampus can encode sequence memory during theta oscillations. It's replayed during sleep in both hippocampus and neocortex in the form of these up and down state or slow oscillation modulated sequence of sequences, that activity can be biased through external sensory manipulation, in this case through the auditory system, and that the sequence memory is paired with reward information during quiet wakefulness and during sleep, but in a different way, suggesting that replay during sleep and quiet wakefulness may serve two different roles. And so you can think of one role learning to actually receive reward, the other role perhaps during sleep, simply trying to evaluate or synthesize model, not based on the expectation that they're going to lead to anything, but just trying to understand the statistics of diverse experience to form models that can then be used to drive reward-seeking behavior. And so with that, I just want to thank all the people in the lab that did this work, including Diane G, who did the early work on the visual cortex, David Foster, who discovered the awake replay phenomenon itself, Fabian Klusserman, Todd Davidson, who did the work characterizing these extended or long sequence replay events, Dan Bender, who did the work in biased reactivation, auditory reactivation of hippocampal memory sequence, and Stephen Gompertz, who is a sort of researcher clinician who did the work on VTA and reward. So with that, I will end it. Thank you.
Oh, that was great. Thank you, Matt. So we have a couple minutes for a couple of questions. So we have one like, is there a way to interfere with the predicted sequences and maybe interrupt like some level of decisions in the animals?
Yes, absolutely. So that experiment actually has been done. So Valerie Ego-Stengel, when she was in the lab, did similar kind of closed loop-like stimulation. That is recording hippocampal sharp wave ripple activity, but then instead of trying to bias it, she introduced electrical stimulation that would disrupt it. And so it turns out if you disrupt replay activity during quiet wakefulness or during sleep, you interfere with learning, and as you get diminished performance. Now, an interesting question is whether you can, there's the sort of bias it, that is bias selection, there's the disruption. The question is, can you engineer them? And that is, can you bias this at the finer time scale? Can you engineer the particular sequences? One interesting aspect of the Bender experiment is that these sequences that were played, the sounds, the left sound, right sound, they were played at random. But if you think about it, you step back, by playing the left, right sequence, Bender was actually imposing or biasing specific sequences. The dream content would have been the animal dreaming left, then right, then right. And so if you think about doing higher temporal resolution stimulation, perhaps you can actually engineer or drive particular sequences, not simply bias sequences that are there or disrupt them.
All right, thank you. And we have time for a last question, I think. So what do you think would happen with this remote extended replay in like more complex mazes or in more sort of like naturalistic behavior situations? Right.
So one thing about this extended replay is that it occurred at basically a fixed velocity. It was just sort of a fixed velocity. And so over, over half a second, the sequences could only be so long. And so kind of thinking about really deep search, could you really think, very far ahead along a sequence? There are two ways of doing that. One is sort of a fragmented sequence. Instead of having it continuous, it could be sort of piecewise discontinuous. You could have, it'd be like thinking of, oh, I'm here, and then I get my car, and then I go to the, the subway station, and I get to work. So there are just, it's a sequence, a route-like sequence. It's not a continuous path in space. And that's generally how we kind of think of solving this really extended sequence problem. The other is to link sequences across multiple sequences or multiple like upstates. So you have like a half a second, that's like a fragment of a sequence, but by chaining those together, and that might be the way in which you evaluate things like the example I gave, the route. And that is, okay, I imagine going to my car, that's like one upstate, let's say a half a second. I imagine going to my car, and then I imagine going to the, the subway station. And so those sequences of sequences could be across subsequent upstates and that would occur naturally during sleep. So you can think of the persistent slow oscillation as being a process of really long, extended sequence of evaluation. And so the idea would be long sequences, you could say extended periods of slow oscillation or extended periods of kind of sequential upstate linkage. That would be the way to improve it, better sleep.
Thank you, thank you. And last question from Sebastián Barrientos. How do you think the sharp wave ripple in the hippocampus recruits cells in distant structures that don't have a direct anatomical connection, like B1?
Excellent question. So, I think, how do sharp wave ripple events, which you can see the modulation downstream, even in the absence of direct coupling. So there are a couple of indirect pathways. So if you actually look within this kind of classic limbic circuit, the hippocampus can talk to, for instance, directly prefrontal cortex, cingulate, retrosplenial cortex. And then those structures in turn can then communicate to primary and secondary association areas. And so the idea is that there are these kind of gateway structures the hippocampus can influence that themselves can produce sharp wave ripple-like activity, and then they subsequently drive these sensory cortical areas. So you have to kind of think about this, this limbic cortical network that might start from the hippocampus but engages these other structures. And not surprisingly, for instance, retrosplenial cortex, cingulate prefrontal cortex have all been sort of implicated in things like mental imagery. And so if you try to imagine, for instance, an extended sequence, like looking at an apple or rotating an object, that engages retrosplenial cortex, and it may involve a similar mechanism biased by the hippocampus, but ultimately it would be retrosplenial cortex that would be driving it in these other areas.
All right, thank you so much, Matt. We have a couple other questions, but in deference to our next speaker, so we're gonna just leave it here. Thank you so much.
Great, thank you, Francisco.
And for our next talk, it is my pleasure to introduce Dr. Dimitrios Milonas. Dr. Milonas is from Greece, and he's an instructor in psychology in the Department of Psychiatry of the Massachusetts General Hospital and the Harvard Medical School. Dr. Milonas is a mathematician who made a radical change in the course of his research and started working in the development and implementation of new analysis methods for neuroimaging data. And currently his research focuses on the relationship between the neural activity during sleep and the impairments of serving learning and memory in people afflicted by neuropsychiatric diseases. Dr. Milonas, welcome, and thank you for being with us.
Thank you, Francisco. Thanks for inviting me, and thanks for the introduction. Let me share my screen. Let's see if that works out. Here. All right, can you see my screen? I hope this works out. So, perfect, thanks. I want to thank you for giving me the opportunity to talk today about the work we're doing in Dr. Manoak's lab in Harvard Medical School. And the message I will try to convey in this talk is that abnormal sleep oscillations in neurodevelopmental disorders are a marker of the underlying pathophysiology, impair memory, and can be treatable. And in this line of work, the focus of our lab has been schizophrenia, but today I also wanted to show some results from the more recent work we are doing in autism. So in brief, I will first introduce sleep in humans and sleep oscillations as the underlying mechanism of sleep-dependent memory consolidation. I will then talk about the relationship with memory consolidation deficits in schizophrenia and some interventions that we are testing to rescue these deficits. And finally, I will go over some unpublished results on what we see in autism spectrum disorder. So with that, I will dive into the first part with a very brief introduction about sleep physiology. So this is how sleep looks like in humans. And this is my partner taking a nap in the car. And although in the outside sleep looks uneventful, it has a structure. And that is reflected in the different sleep stages. This is an example of overnight sleep from a healthy subject. So they go from wake to stage 1, stage 2, stage 3 sleep. By the end of the cycle, they go to REM sleep and the cycle continues. So they go from lighter to deeper sleep. And I won't go into the details of sleep architecture here, but what is important is that these different stages are characterized by different brain activity that is apparent in the EEG spectrogram. Here, the x-axis represent again time, the 8 hours of sleep. In the y-axis are the frequencies from 0 to 30 Hertz. And the colors in this graph represent the power in the different frequencies. And you can see how how slow frequency activity is predominant during stage 3 sleep, which is also called slow wave sleep because it's characterized by this slow oscillatory activity. And you can also clearly see that there's something going on in this band here around 15 Hertz. And this is sigma band and corresponds to sleep spindle frequency, which I'll talk about in more detail in the next slides. But as you probably already noticed, increases in sigma band coincide with the bows of stage 2 and stage 3 sleep. So it's the second cycle, the third cycle, and so on and so forth. So what I want to show with this slide is that sleep is not a unitary phenomenon. It is characterized by different stages during which different oscillatory activity is happening. In the next slide, I will focus on the specific oscillations that take place in non-REM sleep. So this slide shows what we think is the mechanism and the main players of sleep-dependent memory consolidation. And this is a figure adapted from Jan Born's work, showing the 3 cardinal oscillations of non-REM sleep. On top, with the red line, are slow oscillations. These are low-frequency oscillations that originate in the cortex. We can observe them with EEG in humans during stage 2 and stage 3, and this is a cartoon of an EEG electrode and an illustration of a slow wave. So slow oscillations are characterized by steep negative deflection, also known as a downstate. As Matt said before, it reflects synchronous hyperpolarization of cortical neurons and is followed by a positive deflection, which is the excitable state. It's the depolarization of these neurons. And because of the different nomenclatures that exist in the literature, like slow waves, K-complexes, throughout this talk, I'll try to use only the term slow oscillation. An important note here is that although slow oscillations are generated in the cortex, their expression in the brain depends on the interaction between the cortex and the thalamus. And the next oscillation is sleep spindles, here depicted with blue, which defines stage 2 non-REM sleep. Spindles are brief oscillations of 9 to 15 Hertz in humans, and they are generated in the thalamic reticular nucleus, here is in blue, which I will call TRN for the rest of this talk. TRN is a thin structure that surrounds the thalamus, and I'll come back to that later with more details. But spindles are generated in the TRN, but are propagated to the cortex via thalamocortical circuitry. So we can detect them using non-invasive methods like EEG. And this is an example of a spindle, how it looks like in EEG. The last oscillation that completes the picture is sharp wave ripples. And these are high frequency oscillations. They go from 70 to 150 Hertz in humans, and they are generated in hippocampus. Due to the position of the hippocampus, it's very deep in the brain, and the high frequency nature of ripples, we can't record them non-invasively in humans. But we know from work in rodents, and Dr. Wilson spoke just before me, has a major contribution in it, that ripples are associated with memory replay. And in humans, we can gain some insight about ripple functionality from intracranial recordings in patients with epilepsy. What is key here is that these oscillations do not act in isolation. They interact with each other, or they are temporally coordinated, they are coupled. So spindles occur preferentially during the up state of the slow oscillation, the excitable state, and ripples are nested in the spindle troughs. Our working hypothesis is that through this dialogue between hippocampus, thalamus, and the neocortex, newly acquired memories are transferred from the hippocampus to long-term representations in the cortex. And that was a brief summary of the mechanism we think mediates memory consolidation. In the next slides, I will explain how memory goes awry in schizophrenia and the role of these oscillations. And I just want to disclose that I will use we when I refer to work that has come out from our lab, even though a lot of this work precedes me. So schizophrenia is a neurodevelopmental disorder in which symptoms emerge usually in late adolescence, or early adulthood. It's characterized by positive symptoms like delusions, hallucination, disorganized speech, negative symptoms like withdrawal, for example, and cognitive deficits. And although antipsychotic drugs can treat the florid psychotic symptoms, the cognitive deficits remain, contributing to the chronic disability. As a result, approximately 80% of individuals with schizophrenia are unemployed. And I mentioned that to convey the importance of finding ways to ameliorate these deficits. And although cognitive deficits are broad, we focus on sleep-dependent memory consolidation deficits. And we measure memory consolidation using a procedural memory probe called motor sequence task, or MST. Briefly, participants have to type a 5-digit sequence with their left hand as fast and as accurate as possible. So in this example, the sequence could be 4-1-3-2-4. So they have to type with their left hand 4-1-3-2-4, 4-1-3-2-4, and so on, as fast and as accurate as they can. So they complete 12 30-second trials with 30 seconds of rest in between, and the outcome measure is the number of correctly typed sequences. And to give you a picture of what I mean by the memory consolidation goes around schizophrenia, I'll show you data from healthy individuals and schizophrenia patients from a study that came out from our lab in 2004. So this is average data from 20 individuals. During the first 12 trials, they show this nice learning curve. They start typing around 9 correct sequences in the first trial, and after around 6 or 7 trials, they reach a plateau of around 16 to 17 sequences. After that, they go to sleep, and when they wake up in the morning, they perform the same task again. But this time, they start typing faster than before. This performance improvement is sleep dependent. That is, it would not happen after an equal interval of wake. So if they hadn't slept if it was an equal time of wake, their performance would be somewhere there. Now we can easily measure overnight consolidation as a % difference of number of sequences before versus after sleep. And in this case, it would be a 15% increase improvement. But what happens in schizophrenia? So schizophrenia patients are slower in general. Please note here there is a different y-axis on the right that corresponds to schizophrenia, but their learning pattern does not differ from the controls. So they start from an average of 7 sequences per trial, and they plateau around 11 to 12 sequences per trial. So they learn normally. Then they go to sleep, but when they do the task the following morning, they show no improvement at all. And this is an example of process automation. You practice something during the day, and then during sleep, it gets automated. So the next time you do it, it doesn't require as many cognitive resources as before. Failure of this mechanism in schizophrenia can potentially contribute to a wide array of cognition, since it's limiting one's available cognitive resources. And as I said, this was a behavioral study, but after this observation, we brought participants in to perform the MST, and we recorded EEG during their sleep. Again, healthy controls showed improvement in the MST after a night of sleep, but patients with schizophrenia did not. So we looked at spindles during stage 2 sleep, and we saw that patients with schizophrenia showed a significant reduction of spindle density during stage 2. So spindle density on the y-axis here is the number of spindles per minute of sleep. Importantly, these deficits were correlated. The more spindles they had during the night, the more they improved in the MST the next morning, implying that spindle deficit caused the memory deficit in schizophrenia. And although this is just a correlation, you can't infer real causality from that. This causal hypothesis is in line with optogenetic studies in rodents and neurostimulation studies in humans, showing that increasing spindles can improve memory. Of course, a major confounding factor in every clinical study is medication. So to make sure that this was not a medication effect, we brought in early course patients of a wide array and showed that spindle deficits are still present early on. And when we broke down the early cause group into patients that were diagnosed with schizophrenia, with red here, versus other psychotic disorders, the spindle reduction was specific in schizophrenia, suggesting that the spindle deficit is not a function of chronicity of the disease or the medication. Spindle deficits and reduced spindle amplitude are also present in young non-psychotic first-degree relatives. Here the spindle density on the right panel is a spindle amplitude. So they're present in first-degree relatives of schizophrenia patients, leading to the hypothesis that the spindle deficit is a marker of genetic vulnerability to schizophrenia that predates its onset, persists throughout the course of the disease, and impairs cognitive function. But to treat the spindle deficit, we have to understand the mechanism. And there is converging evidence of structural and cellular anomalies in the TRN of schizophrenia patients. Just to remind you, TRN is the structure that generates spindles. And these are results from a post-mortem study in healthy individuals and patients with schizophrenia. On the left, there is an example of a parvalbumin-expressed neuron in the TRN. On the right panel, schizophrenia patients, the red bar, showed a marked reduction in parvalbumin-expressed neurons in the TRN compared to healthy controls. In this study, they also showed that perineuronal nets are reduced in schizophrenia. So perineuronal nets are these extracellular matrix structures that regulate neuronal maturation and protect neurons. So genetic studies support this hypothesis too. A GWAS study in 2004 identified CACNA1I as a risk gene for schizophrenia. This gene encodes a calcium channel expressed only in the brain and particularly in the TRN. Knocking out this gene in a mouse causes specific spindle deficit. So since more evidence is converging to TRN, I have to take a step back and describe its function. So TRN, here with red, is a thin net-like structure located between the cortex and the rest of the thalamus, enabling to modulate thalamocortical communication, sort of gate thalamocortical information flow. So TRN is a major inhibitory nucleus of the thalamus and is completely comprised of GABAergic neurons. During sleep, TRN inhibits thalamocortical neurons, resulting in the rebound burst firing that initiates spindles. Our hypothesis is that failure of the TRN-mediated inhibition reduces the burst firing necessary to initiate spindles and may give rise to spindle and memory consolidation deficit in schizophrenia. Supporting this hypothesis, recent evidence from our lab showed that increased thalamocortical communication might also account for the well-replicated finding of increased connectivity between thalamus and sensory and motor cortex, as measured with resting state functional connectivity MRI. Here with blue are the areas that showed increased thalamocortical connectivity in schizophrenia compared to healthy controls. This is functional connectivity again. And in the same sample, we also found a reduction in sleep spindles in schizophrenia. With warm colors here are the electrodes that schizophrenia patients showed reduced spindle density, and highlighted with green are the electrodes where this reduction was significant. Importantly, TC connectivity, thalamocortical connectivity, correlates with spindle density, consistent with the hypothesis that both measure index the functioning of TRN-mediated thalamocortical circuitry. And that wraps up our view about the underlying circuitry of memory consolidation deficits in schizophrenia. The next step was to target the TRN. So in the next part, I'll go over interventions that we have tested or we're currently testing to rescue this deficit and the lessons we have learned from this. So we used the Zopiclon to target the spindle deficits in schizophrenia. A Zopiclon is an over-the-counter hypnotic drug. We used it because it acts on GABA-A receptor subunits that are preferentially expressed in the TRN, and we did a placebo-controlled crossover study during which we recorded overnight sleep with high-density EEG. We had 58 electrodes. Like before, participants were trained on the MST before sleep and were tested the following morning. So a Zopiclon, as expected, increased spindle density in both controls and patients with schizophrenia. But surprisingly, it failed to improve memory. So in fact, if something, it made memory worse in schizophrenia. So spindles correlate with memory. Zopiclon increases spindles, but does not improve memory. So trying to understand this failure, we went back to the model that I mentioned at the beginning of my talk and realized that we have to look not only at spindles, but also at their interaction with slow oscillations and ripples. And while we can't see ripples in EEG in humans, we could still look at the effects of Zopiclon on slow oscillations and their coupling with spindles. So we went back to our data to detect slow oscillations and measured the slow oscillation spindle coupling. And this is a cartoon of a slow oscillation with a spindle, with this red dot representing a spindle riding on top. The spindle looks like here a point because we use the peak of the spindle, the maximum amplitude of the spindle, to estimate their coupling. So for each individual, we measure the phase of the slow oscillation at each spindle peak, and we do that for all coupled events. So to quantify the coupling now, we can measure the mean or the preferential coupling phase. Here it would be, in this example, on the upstate, on the upstate of the slow oscillation. And we can also measure the consistency of their coupling, which gives us a measure of the distribution of these variants. So in this example, the distribution is relatively tight, so the consistency would be high, whereas in this example, the mean phase would be the same, but the consistency would be very low since spindles are distributed widely along the slow oscillation. So consistency is a measure of how precisely slow oscillation couples spindles. And this is an example of a 30-second EEG epoch with the detected slow oscillations highlighted in green, and an example of a coupled event. Here with green is a slow oscillation, and with red is a spindle riding on the upstate of the slow wave. So going back to our data, we first found that coupled spindle density predicted memory consolidation in both controls and patients, and this prediction was significantly better than spindles alone, suggesting that slow oscillation coupled spindles are a better biomarker for memory. Highlighted with green here are the electrodes where this relationship was significant. And when we looked at the effects of Zopiclon on the other oscillations, we saw that Zopiclon reduced the amplitude of the slow oscillations. And what I show here is a topo plot of the slow oscillation amplitude across all 58 electrodes. Darker colors here show higher slow oscillation amplitude, and you can see the reduction in the Zopiclon condition, and on the right is a % difference. So here blue values mean that placebo was higher than Zopiclon, and you can see that this was a widespread amplitude reduction, significant at all electrodes. So how about coupling? Well, Zopiclon did not change the mean coupling phase, but it reduced the consistency of their coupling with spindles. So made their coupling less precise. And again, that was a widespread effect. Interestingly, coupling consistency was correlated with slow oscillation amplitude. So weaker slow oscillations were less able to precisely couple spindles in their excitable upstates. And this finding suggests that Zopiclon disrupted slow oscillation expression and the thalamocortical dialogue necessary from the precisely timed coupling with spindles that mediates memory. And the lesson we learned is that effective therapies need not only to increase spindles, but to preserve or enhance slow oscillations and their coordination with spindles. The last piece was put together by Matt Wilson and Carmen Varela, where they showed that Zopiclon also suppressed hippocampal ripple power in rats. So you can see here that there is a suppression of hippocampal power after injection with Zopiclon. And this study, although we failed to improve memory, really highlights the importance of considering all three non-REM oscillations when we test treatments for memory deficits. But pharmacological interventions is not the only way to enhance sleep oscillations. And recent work has shown that auditory and electrical stimulation during sleep increase spindles and improve their coordination with slow oscillations. And my colleague, Brian Baxter, recently finished a study in healthy controls where he delivered auditory stimuli in the upstate of slow oscillations during a nap. So we brought in healthy participants. We trained them in the MST. They went into a nap. It was a 90-minute nap where we delivered auditory stimuli, and then they were tested again after the nap. So this is an example here where a slow oscillation was detected online. An auditory stimulus, which is a 50 millisecond pink noise pulse, was delivered on the up state of the slow oscillation. And in this example, it triggered a new slow oscillation and a spindle. So these are some preliminary results showing that auditory stimulation here with red line increases slow oscillation and sigma power compared to no stimulation with a black line. Importantly, stimulation increases the probability to evoke spindles coupled with slow oscillations compared to the no stimulation condition. And on the right hand here, you can see the effect size. And after these results on healthy individuals, we have already started recruiting schizophrenia patients to test whether auditory stimulation works for them. And I have to admit that I'm very excited about these results so far, since auditory stimulation is a minimal risk intervention. And given the wearable EEG devices that have been developed, it could be a scalable treatment to rescue memory consolidation deficits in schizophrenia. And with that optimistic note, I will go to the last part of my talk, which is some more recent work we're doing in autism spectrum disorder. And this is a story that is less mature than schizophrenia, but our motivation for looking at sleep oscillations and their relation to memory comes from evidence that I will show in my next slides, implicating thalamocortical circuitry and the TRN in autism. So autism spectrum disorder is a neurodevelopmental disorder defined by social and communication impairments, along with restricted and repetitive behaviors. Sensory sensitivity and cognitive impairments are also common in individuals with ASD. And despite that sleep disruption is a very common finding in ASD, only few studies have looked at specific sleep oscillations, and the literature is inconclusive. So the first piece of evidence comes from the largest fMRI study in ASD so far, where they showed thalamocortical hyperconnectivity of prefrontal, motor, somatosensory, and temporal cortices in ASD. So notably, this is similar to the hyperconnectivity we see in schizophrenia, suggesting that the failure of TRN inhibition could account for the increased thalamocortical connectivity in ASD. If we look at genetic studies, there is also evidence that genes mediating TRN function may also contribute to spindle deficits and symptoms in autism. For example, PATCH-D1 is a gene selectively expressed in TRN during development, and in humans, over 40% of the cases with deletion or truncating mutations of PATCH-D1 exhibit ASD phenotypes. In mice, deletion of PATCH-D1 gives rise to spindle and attention deficits. So this evidence provided the impetus to analyze data from children and adolescents with ASD in a typically developing cohort. So the data were collected by Maskey and colleagues. That was an overnight study with a sparse EEG array. We had 2 frontal, 3 central, and 2 occipital electrodes. The children were trained on a spatial memory task before sleep. After that, they were hooked up with EEG electrodes. They went to sleep, and they were tested again the following morning. So the task we used to measure memory consolidation in this case is similar to the children games known as memory or concentration. And we chose this task because there are studies showing that the consolidation of this task depends on sleep, and specifically spindle density in young adults, and it's appealing to children. So there were 15 pairs of colored images, and children were instructed to learn the location of each pair. So during exposure, an image was displayed and was followed by its pair. It's an orange in this example. After that, they both were replaced by the squares until all pairs had been displayed. So during training, images were displayed one at a time, and children clicked on the square they thought the other pair was located. If they clicked on the wrong card, an X appeared at the back of the card, and the right location was revealed. So they were trained with feedback until they could recall at least 6 correct pairs. And testing began immediately after training, and the process was the same, but this time it was a single trial and no feedback was given. And after sleep, participants were retested on the same task. So I will go some memory results that were also published in MASCI in 2015. So participants with autism needed more trials to reach the criterion, but by the end of their training, they were at the same level with the controls. They did worse at the immediate test, and we think that is something that it has to do with the lack of feedback, the change of rules in the immediate test. But overnight consolidation, measured as the percent change from test to reset to retest, was not different in ASD compared to healthy individuals. So, as I said, our main goal to reanalyze the data was to look whether sleep oscillations are abnormal in autism. And spindle density and morphology in stage 2 sleep did not differ by group. So black dots here represent data for each individuals, and the bars represent the group mean and the standard error. But spindle density was lower in ASD during stage 3. And to my knowledge, this is the first time spindle density is reported in stage 3 in autism, which in part I think reflects a limitation of visually detecting spindles, a method that many studies in the field have been using. And it also highlights the need to consider non-REM not as a uniform state, but instead take into account that that during different stages, there might be different processes taking place. So when we looked at the relations of spindles with memory, we saw a very different and surprising pattern, different pattern between the groups. So this pattern was similar across electrodes and stage 2 and stage 3 sleep. So I will show here the frontal electrodes. In typically developing children, more spindle density predicted better memory consolidation, a relationship which we expected based on previous studies. But in ASD, higher spindle density correlated with worse memory. And that was very surprising. One hypothesis that we have, and is speculative, has to do with the sleep disruption in ASD. So spindles have been proposed to protect sleep by inhibiting the relay of sensory input to the cortex. And in the context of a general disrupted sleep quality in ASD, higher spindle density may reflect more fragile sleep or a greater need for sleep protection rather than better memory consolidation. So the coordination of slow oscillations with spindles was abnormal in ASD during stage 2 sleep. Here again, black dots represents data for each individual, and the line here is the mean coupling phase for each group. And this is a schematic representation of the slow oscillation. So you can see that spindles peak later in the slow oscillation phase, and their coupling was less consistent, pointing to reduced integrity of thalamocortical circuitry. Spindle slow oscillation coordination was normal in stage 3. And one explanation for this difference between stage 2 and stage 3 could be that high amplitude slow oscillations that are predominant in stage 3 provide a stronger corticothalamic drive and couple spindles more precisely. And supporting this hypothesis, coupling consistency, higher coupling consistency was correlated with higher slow oscillation amplitude, like in our previous study. And although this was not a longitudinal study, the age range of our sample, these were children and adolescents from 9 to 17 years old. So the age range of our sample allowed us to look at how sleep oscillations changed over development. And spindles showed a normal developmental trajectory in ASD, so I won't show that here, but I will focus on the slow oscillations where we had the most interesting finding of age-related changes. So slow oscillation absolute amplitude on the y-axis here, and that is measured in microvolt, decreased with age in both groups. And these data are averaged across electrodes. The decline of slow oscillation amplitude across adolescents is a very robust finding in the literature. and has been proposed to reflect functional optimization of cortical circuits through a process of synaptic elimination, pruning, and rearrangement that increase finally the specificity of cortical connections. But we also measured the relative slow oscillation amplitude. That is, for each individual, we normalized the amplitude of the slow oscillations with respect to the standard deviation of the background EEG activity. And that could give us a measure of how much the slow oscillations deviate from the background activity, or the signal-to-noise ratio. And when we plotted the relative slow oscillation amplitude against age, we saw that its developmental trajectory was very different between groups. So relative slow oscillation amplitude increased with age in typically developing individuals, but declined in ASD. And following the same line of thought as before, the optimization of cortical circuits over adolescents should lead to a better signal-to-noise ratio for slow oscillations, an effect which we observed in the typical developing children. In contrast, the decline of relative slow oscillation amplitude in ASD may suggest a suboptimal cortical reorganization that results in a lower signal-to-noise ratio for slow oscillations in older individuals. And with that, I will summarize. The aim of our research program is to forge empirical links in hypothetical causal chains from gene to diagnosis, with the ultimate goal to identify mechanisms that we can target for treatment. So in schizophrenia, there is an impairment of sleep-dependent memory consolidation. This deficit occurs in the context of spindle reduction, and importantly, the spindle and memory deficits are correlated, suggesting that reduced spindles impair memory. The spindle deficits implicate the TRN and the thalamocortical circuitry, and knockouts and mutations of risk genes for schizophrenia in mouse models impair channel function and give rise to spindle deficits, a hypothetical cascade that may ultimately lead to the diagnosis. In autism, there is also evidence from genetic and fMRI studies that converge to a dysregulation or abnormalities of the TRN and the thalamocortical circuitry, which in turn give rise to atypical physiology. So in autism, memory consolidation seems to be intact, so we need more studies to clarify the functional significance of abnormal physiology, so to establish a link between physiology and cognition. But the point I'm trying to make is that by interrogating these physiological markers with the help of cross-disciplinary research, we may be able to understand better the underlying pathophysiology of neurodevelopmental disorders and bring us to the development of mechanistically targeted treatments. And with that, I want to first of all thank you for coming today. I want to thank my PI, Dara Manoak, who has done a lot of the work I presented today and has supported me during these years. Our funders and that support our research. I want to thank our long-standing collaborator, Robert Strickle, the people who has worked in the lab, and our collaborators in BI, in MIT, in Broad, and at Latin University. So thank you all for coming.
Thank you, Dimitrios.
I finished on time, right?
Yes, perfect. Thank you so much. And that was an excellent talk. But we have a couple minutes for questions. We have a question from Denise Munoz, and she's wondering what kind of electrodes are being used during sleep to improve the memory performance.
So I guess they refer to auditory stimulation, and we use auditory stimulation first of all. So we're not doing transcranial stimulation, and in this case we measure EEG using a high density EEG array. I think we have around 90 electrodes, so we have full scalp coverage.
Perfect. So just auditory, like using headphones, like
using headphones, exactly.
Okay.
Yeah, just like we just deliver like a 50 millisecond of pink noise, like a. It sounds like that.
Got it, cool. And I have a question too. So recently there has some like evidence that the coordination of spindles to slow oscillations may have a differential effect if the spindles are coordinated to like delta oscillations, which are like slightly shorter. Have you guys seen anything like that, or are you planning to like look over that particular?
So this is a very interesting question, and I remember the study by Kim where they showed that delta is related to forgetting, whereas slow oscillations are more enhancing memory. So in humans, it's really tricky to disentangle slow oscillations from delta activity, and the main reason is that the slow oscillations are not sinusoidal, so they have a steep deflection that really leaks into the delta frequency band. So it's very tricky to have objective criteria to disentangle these apart. But yes, this is something that we're thinking about, it how methods of how some sophisticated filtering or modeling to tease them apart.
All right, thank you. And the last question from Benjamin Pizarro is he's asking, how do you interpret the findings in autism within the context of the hypothesis that ASD, schizophrenia, and the bipolar depression are all within the same genetic spectrum? So basically different aspects of the same genetic problem.
Yeah, that's a very good question. And I don't know if I have a clear answer for that, because first of all, these are the story in autism is not so well established. So we're still trying to, draw parallels with schizophrenia. So the same genetic substrate might lead to dysregulation of thalamocortical circuitry in TRN. But that in turn, for some reason that I don't know, give rise to different physiological phenotypes. And it seems like, again, from these results that I saw that are unpublished, that memory consolidation seem to be intact in ASD. And that's also consistent with 3 or 4 more other studies, if I remember well, in autism, probing different types of memory. So that means that maybe autism in autism, there is another compensatory mechanism that mediates or mediates, underlies memory consolidation. But that's a very good question. I don't know if I have a very clear answer for that.
Right, thank you so much. We have a couple of other questions, but in the spirit of trying to keep on time, we're gonna...
I can type the answers in the Q&A.
Okay, perfect. Yeah, that'll be great. Yeah. Thank you so much, Dimitrios. And now we're due for a break. This is going to be, I think, a 15-minute break. So we'll reconvene back here at, say, 11:05. And for the next talk, remember that the questions and answers have to be asked through the Q&A section. And then we'll do our best to answer as many of them as we can. We'll try to keep the talks as short as possible, because as you all probably know, this time in the pandemic, sometimes to make you really tired if you're just watching for too long. So that has been more or less the idea. So also remember that there is an interpretation feature for our talks. So you can either pick Spanish or English interpretation, depending on what your native language is. And I'll see you all back at around 11:05 a.m. So, okay, see you all.
Francisco, I don't hear you. I don't know if you can hear me.
Sí. No. No, no.
Hello, hello.
Yes.
What about now? Yes?
Mm-hmm.
Okay, perfect. Sorry about that. But Zoom Symposium without glitches is not a real Zoom Symposium. So, okay, so welcome back to the second part of this session. And for our next talk, it's my pleasure to introduce Dr. Carmen Varela. Dr. Varela is an assistant professor at the Department of Psychology of the Florida Atlantic University. And her research focuses on the neural mechanism for rule learning and problem solving through memory consolidation and memory retrieval. Dr. Varela also studies the role of the thalamus on the regulation of memory processes and the use of pharmacological strategies to alleviate memory problems observed in neuropsychiatric diseases. Dr. Varela, thank you so much for joining us today, and welcome.
Thank you. Thank you very much. Thank you all for being there, and thank you to the organizers for the opportunity. Let me just share the screen. And let me know if anything is not like looking good, or if you cannot hear me properly or something, so we can adjust at the beginning. Okay, great. So yeah, thanks again. Today I want to tell you about two projects in which we studied the spiking activity in the thalamus and the types of spiking and firing properties that thalamic cells have that we think that contribute to memory consolidation processes. And so in the lab, we're very interested in the acquisition of memory, during which we obviously acquire during wakefulness, but we are particularly interested in what happens to those memories when we go to sleep, during resting states and during sleep stages such as non-REM activity. And so the idea is that during sleep, we are fairly disengaged from the environment that surrounds us, but the brain regions that are important for the acquisition of the memory, they continue to be active and they are temporally coordinated. And we think that it is through these interactions between multiple brain regions that memories are consolidated. And this consolidation process is a complex process, but it's thought to involve at least three mechanisms or three stages in which the memory is stabilized. It becomes more resistant to forgetting over time. And it's also integrated with previous memories that we already have stored in the brain. It's also generalized, so a particular memory becomes useful to the organism beyond the specific experience that originated the memory. Let me just change to the laser here. And so again, we're interested in what happens with memories during sleep, and in particular during non-REM sleep. There are two structures that are key for the acquisition and consolidation of memory. The hippocampus is critical for the acquisition of new episodic memories, but over time, the hippocampus helps the neocortex form a more stable representation of that memory that can be preserved for a long period of time. And it's thought that this interaction between the hippocampus and neocortex is facilitated by the coupled oscillations of non-REM sleep. And you probably already heard about these oscillations early on. In the hippocampus, the one particular oscillation that dominates during non-REM sleep is the ripple oscillation. These are brief, high-frequency oscillations in which hippocampal cells are reactivating previous behavioral experiences that the animal had before going to sleep. And in the neocortex, we also see some typical patterns of activity. And this is just a diagram, but I'll show you some raw data very soon. And the oscillations that characterize the sleeping neocortex include the slow oscillation, which is marked by these downward deflections in local LFPs, and the spindle oscillation, which is a high frequency oscillation, about 10 cycles per second or so. And even though the spindle oscillation can be observed in the neocortex, it actually originates in the thalamus. It is the thalamic local circuits that originate the spindle oscillation in the first place and then engage the neocortical postsynaptic cells. And so the thalamus is thought to be an important contributor to these coupled oscillations that consolidate memories, but only a few studies of the many that have looked into this coordinated activity in non-REM sleep, only a couple of them have looked at the thalamus, and there is much to be known still. And so what I want to tell you about today is two observations that we made, one from extracellular recordings in multiple brain regions, and the other one from some intracellular recordings that sort of complement the extracellular work. And so what I'll show you is how the thalamic activation is fairly sparse during the spindles, and it's actually an interleaved activation such that different cells seem to be active in different cycles of the spindle oscillation. And then in the second part, I'll tell you about our most recent work in which we looked at the burst firing properties in different cells across different thalamic nuclei. Burst firing being important because it's the firing pattern that is key for the generation of spindles. And so starting from with the first project, what we did here was to perform extracellular recordings in multiple brain regions at the same time to study how thalamic cells contribute to the oscillatory rhythms of non-REM sleep. We used multi-electrode arrays to target the medial prefrontal cortex and the CA1 region of the hippocampus in rats, as well as a number of nuclei that are interconnected with these structures. And we asked a relatively simple question, which is how are thalamic cells, how are the spikes in the thalamus coordinated with cortical and hippocampal oscillations during non-REM sleep? And so let me just show you some raw data. This is the local field potential recorded in the CA1 of a rat when the animal is awake, and then when the animal goes to sleep. And you can see how it changes quite dramatically. And during the sleep, we get these transients of activity that are correlated with an increase in firing in the electrodes in the hippocampus. If we zoom in, we can see the typical high frequency brief oscillatory rhythm that is characteristic of the ripple oscillation. This is happening at the same time that there is this increase of activity. These are the cells that we think are reactivating previous memory traces, previous experiences that the animal had before going to sleep. So that's what happens in the hippocampus. In the cortex at the same time, if you look at the LFP of a sleeping rat, this is the typical oscillatory patterns. There are these downward deflections every now and then, which correspond to a decrease in the local spiking activity. These are the down states of the slow oscillation. And we also see higher frequency patterns such as these here. These are spindles, and you can see them a little more clearly in the filtered LFP. And so I told you that these oscillations are coupled. What this means is that they occur close in time with each other. And it's easier to see that if we overlay them, if we just plot them in the same plot, which is what I did here. And I'll just focus on two of the traces that we're seeing here, just for simplicity. The black line in the background is the ripple filtered LFP recorded in CA1. You can see these blobs of black, they correspond to ripples happening in the hippocampus. And now we are seeing the overlaid LFP from the cortex, and the blue one, the blue trace, is the spindle filtered LFP. And so I think you can see fairly clearly how ripples tend to occur nested in the troughs of the spindle oscillation. These are structures or LFPs that are recorded in different structures which are far away, but still they coordinate their activity down to the millisecond level. In fact, if you detect all of the spindle troughs recorded through sleep, and you plot a peri-event histogram of when ripples happen with respect to those troughs, you can see here how there is a clear increase in the ripple rate at the time or around the time of the troughs of the spindles. Now, how does this matter? How does it matter that ripples are nested in spindles? Does it have any relevance for what the animal did when he was awake? And so what we did was to have some of our rats explore an 8-arm radial maze for about 30 minutes or so. And at the time that the animal was exploring, we recorded the spike activity of cells in the hippocampus. Each dot here is a spike. So these clusters represent spikes from different cells in the hippocampus. And then we kept track of those cells as the animal then went to sleep after this behavioral task. And what happens is that often you record more cells during sleep because not all of the cells that are recorded by one electrode contribute to the representation of a particular behavioral experience. And this is convenient for us because then we can compare the cells that were active in REM to those cells that were not active when the animal was awake and see how they relate to the different oscillations. And so what we found was that cells that had been active in REM before the animal went to sleep, when they produced the spikes in ripples nested in spindles, they tended to fire those spikes at specific phases of the spindle oscillation recorded in cortex. This cell here is firing most of its spikes around the trough or the ascending phase of the spindle oscillation in the neocortex. And instead, cells that were not active in REM, they didn't care so much. They didn't have such a strong preference for the phase of the spindle recorded in the neocortex. And just to show you the summary for all of the cells that we recorded in different animals, cells that were active in REM, they phase locked to the spindle oscillation when they fire the spikes in ripples that were coupled to spindles, but they didn't care so much about the phase when they fire the spikes in ripples that were not coupled to spindles. And we didn't see a particular difference for cells that were not active in REM, which didn't care so much for the phase of the spindle oscillation. And so what this says is that when ripples are coupled to spindles, the cells in the hippocampus use the phase information of the spindle to correlate with what's happening in the neocortex. They tend to be temporally aligned with things that are happening in the neocortex. And so going back to the original question, what's then happening at the level of the thalamus? What are the thalamic cells doing at this time? Well, here is a spindle recorded in the thalamus. And one thing that was a little surprising was that the cells, individual cells in the thalamus were not very active. They were very sparse. And if we ran autocorrelograms, which if a cell is rhythmically active during an oscillation, they will have a clear peak. We didn't see clear peaks with those cells. And this is not to say that the thalamic activity is not contributing to the spindle oscillation. If I show you all of the spikes from multiple cells recorded in the thalamus, you can see how every cycle of the spindle oscillation has a few spikes in the thalamus, but they are not coming all from the same cell. And in the autocorrelogram of all of the cells, you do see peaks that indicate that the overall multi-unit activity, the population spiking, is occurring at about 10 Hertz. And so this sort of brings up two potential models of how the spindle generation is being generated. You could think of a rhythm as being generated by individual cells firing in every single cycle. However, that is not what our data suggests. Our data suggests that individual cells are fairly sparse in their contribution to spindles. They only fire spikes occasionally in some of the cycles, but you still get spikes in every cycle because other cells will be contributing spikes in different cycles of the spindle oscillation. In other words, there is an interleaved activation of different cells through the spindle. And so this type of model also brings up the possibility that perhaps even if a cell is not particularly active in all the cycles, maybe once it activates, it consistently activates with other cells. And so to answer that question, we did cross-correlograms between pairs of cells in the thalamus recorded at the same time. And in fact, we saw some evidence that cells tend to co-activate with other cells. They are fairly sparse, but if you run a cross-correlogram, such as in this case, and take the spikes of one of the cells as reference, the spikes of the other cell, they occur preferentially within a 50 millisecond window or so, even though none of the individual cells was particularly rhythmic in their firing. And so this suggests it provides some support to this sparse synchronicity model in which the spindles observed in the neocortex are generated through the sparse activation of thalamic cells, which are co-activated or tend to activate close in time with other thalamic cells. And let me just summarize this and give it a little more context before we move to the other second part. And so what I showed you basically is how ripple activity in the hippocampus tends to occur nested in the spindles of recorded in the neocortical networks. And when cells that have been active during wakefulness fire spikes during those ripples, they tend to align their spikes to specific phases of the spindle oscillation recorded in cortex. Instead, cells that were not active during behavior, they do not care so much for the phase of the spindle, even though they still activate during ripples that occur nested in spindles. And so at the same time, the thalamic cells are very sparse in their activation. Different cells are active in different cycles. And once a cell activates, it tends to co-activate with other thalamic cells. And so we think that this might be important for consolidation processes, and in particular for the integration of memories from the hippocampus into neocortical networks with other memories that are already stored in the neocortex. And this is because every time that you have a ripple in the hippocampus, it may find a different subset of thalamocortical cells that is being active in different cycles of the spindle oscillation. And so let me then give you another snapshot of like some of the work that we are doing here in my lab. And this, in this project in particular, we looked at burst properties in different thalamic cells. And this is because burst firing is a main firing mode in the thalamus during sleep. It's the predominant firing mode of thalamic cells during sleep. And it's also a firing mode that it contributes to the spindle generation. And so we wanted to know if there are properties of burst firing that maybe could account for this sparsity of burst firing and activation during the spindles that we saw in the extracellular study. And so just to give you a little more background, basically thalamic cells can fire in two fundamentally different firing modes. When they are relatively depolarized, which is what happens during wakefulness, they fire spikes in tonic mode. These are the typical action potentials, sodium potassium action potentials. But when thalamic cells are hyperpolarized, which is what happens during sleep and resting states, they fire spikes predominantly in burst mode. This is because hyperpolarization activates a transient calcium conductance that these cells have that makes them fire spikes, a brief burst of spikes in quick succession. And this burst firing mode is quite interesting because it's observed across cells in all of the thalamic nuclei. It contributes to sleep oscillations, and it's thought to be a signal that is fairly standard across cells in the thalamus. But we wanted to see if that was really the case, how stereotypical these bursts are. And so to answer that question, my research assistant Nidhi Desai, she worked on a data set in which we had intracellular recordings of bursts in different cells across several thalamic nuclei. In this data set, which was acquired in brain slices, rat brain slices, we had recorded bursts in 6 thalamic nuclei. And the names are not really going to be critical here, but the main point is that we had 6 thalamic nuclei from 3 different sensory systems: visual, auditory, and somatosensory. And 3 of the nuclei were from first order parts of the thalamus. These are the parts of the thalamus that receive the input from the periphery, from the external world first, such as the LGM, which gets input from the retina. And then 3 of them were higher order nuclei. These are nuclei that receive input from cortex, and they are higher in the thalamocortical hierarchy of the different systems. And so this allowed us to compare bursting properties in different functional components of the thalamocortical network. And so what we did in these experiments was to inject negative current pulses to induce bursts at different hyperpolarization levels. And basically Nidhi studied a number of properties of those bursts, such as the latency to produce the first spike and the number of spikes in the burst. And so here is our first result. So the first thing that we noticed that was that bursts that were producing higher order cells, they tended to have more spikes in the burst. And so what you're seeing here are bursts from 6 different cells recorded in the 6 different nuclei. And all of the higher order nuclei cells have many spikes in the burst. But the first order nuclei were a little more variable, and the visual and the auditory cells, they tended to produce very few spikes in the burst. And let me show you the population data for these. The distributions of spikes per burst overlapped quite a bit, but overall we found that cells in the higher order nuclei were more similar, had more similar distributions for those nuclei, and they tended to produce more spikes per burst. And instead, the nuclei in the first order thalamus, they were more variable, but the visual and the auditory system produced bursts that had only 1 spike on average. And so this is interesting because the number of spikes per burst actually can have an influence on how thalamic cells engage the postsynaptic cells in the cortex. Now, another interesting finding was that we also saw differences in the latency to produce those bursts. And so here I'm showing you 2 bursts from 2 cells in the auditory thalamus, and both cells are hyperpolarized to a similar hyperpolarization level, but the cell in the higher order thalamus takes much longer to produce that initial spike in the burst compared to the cell in the auditory thalamus. And here is another example, this one from the somatosensory system. And again, the same thing, even though the cells start at about the same membrane potential, the cell in the higher order thalamus takes much longer to produce the first spike in the burst. And so overall, for the latency to produce that first spike, we found also that the higher order cells were more similar, the distributions overlap, but in general, the latencies were much longer in higher order nuclei and more similar across the 3 sensory systems, whereas the first order nuclei were more different between themselves, and the auditory and the somatosensory thalamus for the first order tended to have much shorter latencies to produce the first spike in the burst. And so we then looked at a number of things, a number of properties in these cells to see if there was anything that could explain the variance, the variability in burst properties that we were seeing across the 6 nuclei. We didn't see any difference in the age of the animals or in passive properties such as the membrane capacitance or the membrane resistance, or in other conductances such as the presence of the H current, which is also activated by hyperpolarization. Occasionally, we saw a nucleus that was a little different in their properties with respect to other nuclei, but in general, we didn't find anything that explained the different burst properties that we were seeing across the 6 nuclei of the thalamus. And then we started wondering if maybe it could be the properties of the T channel itself that could induce different burst properties across different nuclei. And so to study that, we implemented a single compartment model of a thalamic cell, which incorporated all of the conductances that are typically found in these thalamic cells, including obviously the transient T current. And we ran simulations using the Dynasim simulation software environment. And this model replicates the tonic firing and also the burst firing that is typical of thalamic cells. And then we changed the properties of the T conductance in the model to see which ones could influence the burst properties as observed in the intracellular recordings. And so what we did was to systematically change the amount of current that flows through the T channel. And we also changed the voltage dependence of these conductance. And this is because normally the T current is activated for hyperpolarized in hyperpolarized potentials, but we wonder what would happen if these the activation and the inactivation properties of the channel were to be shifted towards more depolarized potentials. And so here is what we have. And just to like walk you more easily through this table. So when we didn't change the voltage dependence and we only increased the amount of T current, which is what you're seeing in the first column, we saw that the number of spikes per burst went up. And so increasing the current leads to an increase in the number of spikes per burst. But it didn't really change the latency much. And so it was only when we both increased the amount of current and when we changed the voltage dependence of that conductance that we started to see burst properties that corresponded to those that we saw in the intracellular recordings. And so this suggests that the higher order nuclei of the thalamus may have T current, a T channel that has a slightly different properties than the channels that mediate the current in first order nuclei. And so to summarize this data set before we go into one more point about the implications of these different burst properties, what we did was to study burst properties in 6 nuclei of the thalamus. They were all from the sensory thalamus, from 3 different systems, 3 of the nuclei from first order regions, and 3 of the nuclei from higher order areas of the thalamus. And we found that overall, the higher order nuclei are more similar between themselves. They have bursts that on average have more spikes per burst, and they also have longer latencies. And instead, the first order nuclei were more variable in their behavior. It seemed like for the first order nuclei, it was the sensory modality actually made more of a difference. And so cells in the visual first order nucleus had burst properties that were different from all of the other first order nuclei. They only produced 1 spike per burst on average, and they had longer latencies. And instead, the somatosensory first order part of the thalamus had much a stronger burst, if you want. They had many more spikes, and the latency was much shorter, and the auditory thalamus was somewhere in between. And so the implications of these findings are important, first of all, for thalamocortical transmission, because having more bursts per spike can activate the postsynaptic cells more strongly, and we did some simulations related to that in the paper. But I wanted to bring up another implication, which is going to connect also to the oscillatory activity during sleep. And that is that the latency to burst can condition and can determine the oscillatory frequency at which a cell can fire bursts. And so it can influence the oscillatory properties such as those during non-REM sleep. And so what we did to test how different cells could or could not keep up with different frequencies was to use the model to inject pulses, negative current pulses, to induce rebound bursting at different frequencies. And we asked the question of if we impose on the model different latencies, can that model keep up with different frequencies? And so this is the basic setup in which we injected negative current pulses at slow frequencies, and then we progressively increase the frequency to try to force the compartmental model to produce bursting at higher frequencies. And so what we did is that we ran this experiment or this simulation with 3 different types of latency. And so what Nidhi found was that if she had the model with a latency that is more similar to what we found in first order nuclei, then the model cell was able to keep up with frequencies up to about 10 Hertz, and in some cases up to 15 Hertz. And so these are the frequencies of the spindle oscillation. And instead, like if we had a model cell that had latencies to burst that were more similar to what we found in the higher order nuclei, these cells, the model cell was then very limited in which frequencies it could follow. And it could keep up with the slower frequencies up to about 6 Hertz, but beyond that, it could not produce bursts at higher frequencies. And so that suggests that these latencies may actually provide or constitute an intrinsic mechanism that limits the oscillatory rebound frequency properties of thalamic cells. And which can have implications for sleep oscillations, because the cells with the longer latency to burst may be more tuned to the slow oscillations and may not be able to keep up with the higher frequencies of the spindles. It could also help explain, or partly explain at least, why we were finding such sparse activity in the thalamic cells that we recorded in the other study, which were mostly higher order nuclei. And so let me just summarize the two projects, basically coming back to where we started. So hopefully this makes more sense now. Basically, in this initial study in which we used extracellular recordings in multiple brain regions, we found that there is a sparse and interleaved activation of thalamic neurons during sleep spindles. And we think that this may contribute to consolidation processes, because it may activate different thalamocortical cells in different cycles of the spindle oscillation, perhaps to facilitate the integration of the reactivated memories in the hippocampus into memories that are already in the neocortex. And the other finding from this other study, in which we used intracellular recordings and compartmental modeling, is that the burst firing properties that are generally thought to be similar across thalamic nuclei, they may actually vary depending on the modality of information that a nucleus is processing, and depending on the hierarchy in which that nucleus is embedded. So in general, what we found was that the higher order nuclei, those that contribute to cortical, they receive input from cortical regions and contribute to cortical processing of information, they have more spikes per burst, and they have slower latencies that may make those cells be more sparse during spindle oscillations. And the first order nuclei were more variable in their burst properties, suggesting that the modality or the type of information that is being processed may be a more important determinant of the bursting properties early in the hierarchy of the thalamocortical systems. And I just want to finish, and I think I'm running, probably talked quite fast, because I think I'm going to have a few extra minutes for questions, which is great. But I want to first acknowledge everybody that helped me with these projects. The first project was done in Matt Wilson's lab when I was a postdoc there. The second project was led by Nidhi Desai, a research assistant in my lab. These are the other lab members, and we are a growing lab. We are only 2 years old, but we are growing. So if you're interested in sleep, memory, or thalamic networks, and you're looking for a new position, please do send me an email and we can talk more about this, and also the funding sources. And I'll open it for questions. And thank you everybody for attending.
Thank you so much, Carmen, for that excellent talk. Also, don't forget to mention that the lab is located right next to the beach, so that might also help with the recruitment.
Sunny and
yes. Okay, so we have a couple of questions. One from an anonymous attendee is asking, like, if the changes observed in the higher order nuclei can be due to a change in the unitary current of the T calcium channels.
In the what? Say that last part
the
again.
Unitary current of the T calcium channels?
Yes, definitely. Yeah, exactly. Absolutely. So there are, I guess, upcoming questions, right? Like one is exactly that. That's one possibility. Another interesting possibility is that there are some results from genetic studies that suggest that the 3.3 channel, there are 3 T types or T-type channels expressed in the thalamus, and the 3.3 seems to be expressed to at least a little extent in some higher order nuclei. And so it's possible that those nuclei have this additional channel, which is known to have different properties. But yeah, you're absolutely right. The conductance or unitary current of each specific channel could definitely be a factor here as well.
Okay, and within the same question, did you notice any difference in the after depolarization of the different thalamic nuclei?
In the after depolarization, so we did look into that a little bit, but we didn't see, so there is some variance, but we didn't see anything consistent with this idea of functional architecture. So we were not able to relate it specifically to either the sensory modalities or to the first order, higher order hierarchy in the thalamus. So there might be something interesting there, but it might not be explained by the factors that we looked into.
Okay, another question by Elias Leiva. So is there an indirect effect of the calcium currents mediated by the T-type channels in the modulation of the latency or the timing of the thalamic depolarization?
Yeah, great question. So we didn't look at that directly, but we sort of looked at that by looking at the H current. So the H current is a mixed cation conductance, which will, let calcium through and sodium. And we didn't see, again, like we saw some correlation, and actually the more of the H current that was present in a cell, the shorter the latency, but that was true regardless of the functional architecture again. So there are some interesting dynamics with, calcium potentially coming in through these other conductance and what happens with the T current itself. And, but again, like it doesn't explain the variance or the different properties that we saw in the different nuclei.
Okay, I see. And the last question is something that occurred to me. So the thalamic neurons were like during sleep or like states of like hyperpolarization, not only firing in bursts, but also those bursts tends to occur like rhythmically, typically at 4 hertz, 3 hertz, kind of like a delta oscillation. How does that like intrinsic thalamic oscillation gets coordinated with like the spindles and the TRN and thalamocortical system? Yeah.
Yeah, that's a fantastic question as well. I mean, I think it's a population. We have to think about like the population dynamics to fully answer that question. And I think if anything, the results that we are finding, they suggest that maybe different parts of the population contribute more to some oscillations than to the others. Like some of them contributing more to the delta slower oscillations, and some of them more to perhaps the spindle oscillation. But something has to bring them together, right? As you're saying. And so the interactions with TRN and the interactions with the cortex, I think that's what's going to be important to determine that the cortical slow oscillation has a strong influence also on the thalamus, which can be entrained into that slow oscillation. So it might be one of the sort of masters to like bring everybody into the rhythm, but that's population dynamics, right? I think that's the key.
All right, thank you so much. Thank you. Excellent presentation. And for the last talk of the day, we welcome our own Dr. Elias Leiva Salcedo. Dr. Leiva obtained his PhD from the University of Chile and did his postdoctoral training at the National Institute of Health in the United States. Dr. Leiva is currently an associate professor in the Faculty of Chemistry and Biology of the University of Santiago de Chile, and his research focuses on the role of ionic channels and currents on a variety of cellular processes, and specifically in the role of the TRIP channels in sustained neural activity, which is thought to be a cellular correlate of working memory. Dr. Leiva, please, and welcome.
Profesor, no podemos escucharle.
Okay. Can you hear me now?
Yes.
Sí.
Thank you. Okay, today I will talk about the mechanisms of persistent firing, and I will show you some of our results in the lab. Okay, so we know that when two neurons connect together through synaptic connections, these synaptic connections are always changing, and long-term changes appears to be involved in the memory formation or long-term memory formations. However, at rapid time scales, there are really profound changes taking place in the neurons. And the question is how these changes are related to presynaptic activity, and how this presynaptic activity generates a diverse variety of postsynaptic activities, such as burst firing, non-regular spiking, or regular spiking. To explain this, we have two ubiquitous mechanisms to explain this kind of persistent firing. And one is the changes in the intrinsic excitability of the individual neurons, or the other is the change or the activation of reverberant circuits on recurrent networks. So what are the functions of persistent activity? First, it was described, this activity, in several areas of the brain, but one of the most known activities is the persistent activity during the delayed response memory task. Okay, when an animal is presented with a cue, visual cue, and they are and the animal have to remember where the cue was, between the time between the presentation of this stimulus and the recall of this memory, there is a delay, and some neurons spikes at this time with no basically sensory stimulus. Okay, the same effect is observed also when animals follow an object and move their head and change direction, as you can see here. When the animal moves, there is an increase in this spike or firing rate. And this is correlated basically with an increase in the firing after the stimulus is presented. So what are the basis of this persistent firing? Basically, synaptic reverberation in recurrent circle consists in the activation of neurons between each other, okay, in circuits, and one neuron activates another, and this is successive rounds, and they start to firing in a persistent way. The other is the modulation of the intrinsic excitability, in where the diversity of channels expressed in different areas of the neurons can make account or take account of this kind of firing patterns. Okay, we know that today that a combination of both mechanisms makes account of the persistent firing. So in reverberant circuits, one neuron activates another, and the other, and the other, and you have the persistent activity. And the question is how this is stopped. And generally, this is stopped by a strong stimulus that can hyperpolarize the neurons or block the transmission between the neurons. Okay, but how these neurons accumulate this signal in the, sorry, let me a little bit. Can you see now my presentation?
Yes.
Thank you. So how the neurons accumulate and store signal is the question, because neurons in this model can accumulate and maintain signal for several seconds. When you apply a stimulus to the neurons, and if the neuron behave in this way, it starts accumulating the signal and makes the neuron to enter in an app state where the persistent activity is easy. So to take the neurons out of this state, you have to give to the neurons a strong stimuli. Okay, but in the reality, neurons intrinsically have brief periods of activity. So you have stimulus that are restricted in time, and the neurons follow this stimulus by activity in a restricted period of time. So the question is how neurons can sustain this activity. And we know that repetitive stimulation of inputs inducing persistent firing always led to a well-defined increase in the stable discharge rate of the neurons. And once the neurons enters in this persistent firing, it could only be turned off by prolonged membrane hyperpolarization. Also, the repetitive application of hyperpolarizing current pulse leads to a graded stable decrease in firing rate. So could local synaptic activation also lead to a state of persistent firing? This is the question. Okay, so persistent firing can be expressed in several different ways. Okay, so you have here a classical response of a neuron where you stimulate the neurons, you have the action potentials, and then you turn off the stimulation and the neuron is silent. And again, the activation and the neurons stop the firing. But when you have graded potentials, you can have a kind of accumulation of this depolarization, and you have that when you increase the stimuli, the neurons reach higher levels of probability to fire. And this can be achieved by several type of current, including the calcium-activated non-selective cation current, or ICANN, say that there. Also, you have intrinsic oscillations in the membrane potential that make the resting membrane potential change in time and make it more probable to fire action potentials in certain areas of these depolarizations. And also you have the plateau potential that can increase the depolarization in a steady way, okay, and increase the probability of firing action potential for long periods of time. And several currents have been involved in this this type of mechanism, such as calcium-activated non-selective cation currents, and also sodium persistent currents. In this way, the depolarization of the neurons, particularly in this graded depolarization, in some neurons increase the firing probability, okay, and through the plateau potential. And this has been described for an effect, sorry, for the activation of the sodium persistent current, which you have here in this graph, where its activation, in this case by acetylcholine, you have the activation of the current that keeps the neuron depolarized and making them fire action potentials in a regular manner. Okay. So, but what happened with another kind of channels? Okay, this ICANN current, which has many members that has been proposed to be the responsibles, are calcium-activated cation currents. This current can be activated by several types of stimulus, including the activation of neurotransmitter receptors, the voltage-gated calcium channels, and other calcium sources. And what happened when the calcium increase in the intracellular neighborhood of the channel, this can activate the current, and then this current depolarizes the neuron, and this depolarization increase the probability of firing, and then because the depolarization decrease the driving force for calcium, the neurons stop to firing because calcium returns to their normal levels, as you can see here, between the changes in the calcium concentration, the driving force, and also in the membrane potential. So this produces a regenerative wave of calcium that can keep and sustain the activity of this channel. Okay? And this can generate this persistent activity. In this way, TRPC4 and TRPC channels, which are two channels of the family of TRP channels, they have been involved in this type of fighting, and as you can see in this experiment from the Arboid and collaborators from 2010, you have that the application of an antibody against TRPC4 stop the persistent fighting when you apply in the bath, and the same with TRPC5. While these two channels are not canonically described as a CAN channel because they are not directly activated by calcium, they have some similitude to other TRP channels that can keep this activity. In our laboratory, we determined that this activity, the persistent activity, is related to the expression of an after depolarization, which is a membrane depolarization occurring just after the repolarization of the membrane potential during the action potential, and this depolarization increases the probability to fire action potentials, and the more depolarized, the more probable is to have an action potential after this. And as you can see, when we inhibit TRPC5, we lost basically most of the after depolarization, and we observe the stop of the spiking of the neurons. Okay, so we know that all of these activities are related basically to circuits and the proteins that the neurons participating in these circuits, which proteins are expressed in these neurons participating on these circuits. Okay, so we know that neurons express differentially channels and receptors through their structure, and also particularly in the cortical area, in different cortical layers, neurons express different receptors, different channels. So through the modulation of these channels, you can obtain very different responses in the neuron. Okay. So our laboratory focused on investigate the role of this, the channels, the localization of the channels, and how they participate in the modulation of neuronal firing. So we know that a CAN channel, TRPM4, which is a calcium-activated, non-selective calcium channel, calcium-activated channel, sorry, that only permeate monovalent cations, okay, is expressed in the prefrontal cortex, particularly in the layer 2 and 3, yeah, and it presents a very particular type of expression where it's mostly somatic and in the proximal dendrite, but is absent from the axon initial segment, okay? And this channel, when we record neurons that spike spontaneously, when we inhibit these neurons, these channels, or in these neurons, we have a decrease in the firing rate. So this indicates us that these channels, it is participating in the firing or in the repetitive firing in the neurons. So we tested also if this is also true for stimulation induced by different neuromodulators, such as in this case carbachol, which is an analog of acetylcholine. And you can observe that neurons spontaneously fire, but when you apply the carbachol, you increase the probability of firing, but in the presence of this inhibitor of TRPM4 called CBA, you have a decrease in the firing. Okay, and this firing also is this decrease in the firing is also observed in neurons that express an shRNA against TRPM4. That when we record this from slices in the layer 2-3 of the prefrontal cortex, we observe a decrease in the spontaneous in the frequency of the spontaneous firing of these neurons. Okay, so what are the bases of this, or what is the mechanism that is sustained on this channel? The question we started to looking before is, was TRPM4 doing something in the resting membrane potential or in the basal current of the neurons in the layer 2-3 of the prefrontal cortex? And we observed that when we inhibit the channels, we have a hyperpolarization of the resting membrane potential, which is correlated with a decrease in the basal current of these neurons. So this suggests that the channel is open at the resting membrane potential, and it may be participating in the setting of the resting membrane potential. So because this is a calcium activated channel, this implies also that the calcium inside the cell is enough to keep the activity of this channel. So this channel must be really, really sensitive to calcium. And we know that these channels can be activated by concentration of calcium as low as 50 nanomolar of calcium inside the cell. So we investigate deeper this effect, and because of the expression of the channel that we observe in our immunofluorescence, we start to looking at where the channel have its main effect. So we use this setting where we record the neurons and we locally perfuse the inhibitor of this channel in different areas of the neurons. And you can observe that only when the inhibitor is applied in the somatic region, we obtain a decrease in the currents with no stimulation. So again, the channel is active at resting membrane potential, and most of its activity is restricted to the soma. Okay, this is also similar in CA1 hippocampal neurons, where the channel is also expressed in the soma, but also in the dendrites, in the dendrites of the CA1 neurons, and also is active at resting membrane potential. Okay, so you can see here the perfusion of the inhibitor reduce the current, and also this current is activated by calcium, since when we decrease the intracellular concentration of calcium using EGPA, we observe that there is almost no current, and this current also induced is open because when we apply the inhibitor, the neurons hyperpolarize, and also the input resistance of the neuron is increased, indicating that basically the channel is open at this the resting membrane. Okay, so this is the effect in resting condition. But what happened when we stimulate the circuit? So to do this, we perform this experiment where we apply a high frequency stimulation, 100 hertz for rounds of the stimulation in the layer 1, and we record neurons in the layer 2 or layer 3 and observe the effect of the pharmacological inhibition of TRPM4. And you can see here that when we apply this stimulus, we observe an increase in the firing frequency that is basically reduced when we perform the same protocol in the presence of the TRPM4 inhibitor. So this suggests us that TRPM4 participate in this, increasing firing rate. Also, when we perform the same experiments in slices transduced with the shRNA against TRPM4, we observe that at basal conditions, there is a reduced firing frequency, and also we didn't observe any doublet or triplet firing that is common in the, as you can see in the scramble transduced neurons. And also we observe a lower effect of the high frequency stimulation in the firing rates. Also, we observe that the resting membrane potential is more hyperpolarized in neurons that express TRPM4 shRNA. But what is doing TRPM4 in this particular effect? So, because of the doublet firing and triplet firing that we observe in these neurons, suggests us that something was happening in the after the polarization. We perform protocols to activate just one action potential, and we observe the development of the after the polarization. And as you can observe here, high frequency stimulation increase the amplitude of the after the polarization, and this is reduced when you pharmacologically inhibit TRPM4. Also, when you decrease the concentration of calcium in the cell, you observe the same effect, that a reduction in the amplitude of the after the polarization. So, what happened when we reduce basically the expression of TRPM4 using this shRNA? We observe basically a decrease in the ADP and almost no effect of HFS, high frequency stimulation, in the after the polarization, indicating us that TRPM4 play a critical role in the after the polarization, and suggesting us that this may be the mechanism that is increasing the firing of the neurons. Okay. So, because calcium is critical for this effect, we also tested another types of stimulus that can increase the firing of the neurons and increase also the intracellular calcium. And we use the mGluR type 1 receptor, which can activate the GQ copper protein receptors and increase the intracellular calcium. We use DHPG, which can activate this receptor, from a drug that can activate this receptor. And you can observe that DHPG increase the firing, but in the presence of this TRPM4 inhibitor, the increase of there is a reduction in this firing. So this suggests us that TRPM4 appears to be playing an important role in the calcium-mediated increase in the firing frequency. Okay, so the conclusion of our experiment are that TRPM4 is critical for the sustained activation of action potential firing. Also, that TRPM4 is active at resting membrane potential, and it's participating in the setting of the resting membrane potential. And also that TRPM4 expression is restricted to particular areas of the neurons, and this can have several important implications in the circuits and how they are modulated in the circuits. And also that TRPM4 is responsible after depolarization, and this could be the basis of how TRPM4 increase the firing frequency in these neurons. So we have this working model, which is, we have several question marks and how is this system is activating TRPM4 and how is modulating the firing frequency. But in general, we think that receptors for neuromodulators or neurotransmitter can in some way increase the intracellular calcium through intracellular reservoirs like endoplasmic reticulum, or through voltage-gated calcium channel. This calcium can activate TRPM4, which then depolarize the neurons and increase the firing frequency of these neurons. Also, in similar ways, the activation of ionotropic glutamate receptors can increase the intracellular calcium in the neighborhood of TRPM4, or activate through the polarization the voltage-dependent calcium channels, and this can be providing the calcium necessary for the activity of TRPM4, which, again, once it's activated, it can depolarize the neuron and increase the probability of firing of these neurons. So finally, I would like to thank the people in my laboratory, which perform most of this work, Denise Riquelme and Francisco Peralta, also Nicole Machuto, and Franco Navarro and Claudio Moreno, who perform most of the experiment in this presentation. And thanks for coming.
All right, great presentation. Thank you so much, Dr. Leiva. And we do have a couple of questions here. First one is from Ignacio Ramirez, and he'd like to know if just the alkaline cations act on the membrane potential, or other metals also like copper or rhenium could have an effect.
Okay, this is a good question, particularly in the case of these channels. There is no evidence that this kind of metals can modulate the activity of the channels. However, in the case of other channels like P2X receptors, which are activated by ATP, several metals can modulate their activity. So, for instance, P2X7 and P2X2 or P2X4 have a high sensitivity to copper, for instance. So, and I don't know if P2X4 or P2X4, I think, is not expressed in the cortex, but at least P2X2 and P2X7s are expressed in the cortex, so it can have some role.
Okay, cool. I have a couple of questions too. So you mentioned that the TRP channel is present in prefrontal cortex and in hippocampus so far for what. So do you think that hippocampal prefrontal coordination is also sort of like fundamental for the memory consolidation, from transfer from hippocampal memory to like cortical memory? Do you think that these channels could actually have a role on facilitating that process?
It's an interesting question, and we think that it can participate in the recall of memories through the working memory. Okay, so in this way, and it can be participating in the persistent firing that is induced during this delay period between the recalling of the memory or the sensory stimulus. But I didn't mention it in the presentation, but there is a great debate about the role of persistent firing in this delay response in the working memory. So there is a great the two articles in Journal Neuroscience 2 years ago from the Miller's lab and from Constantinidis' lab discussing whether the persistent activity observed in the delayed period is an artifact or is the basis of the working memory. So there are basically evidence for both. So you have to choose, basically.
Okay. I guess that will give you more work to do in the future.
Yeah, there is. Yes. But the persistent firing in this case, in this type of function, is in debate. But persistent firing is manifested in several other functions, not only in the delayed period during working memory tasks.
Okay. So something also that it wasn't like entirely clear to me, like for example, a reverberant circuit that is just like active for too long, especially if it's like pyramidal network neurons, could constitute like an epileptic seizure. So what are the classical like mechanisms to stop that from happening in the brain?
Okay. That's a good question, and I didn't go through in deep, but in recurrent circuits, if you think only in pyramidal neurons, okay, it's almost impossible to stop, okay? But in the recurrent network, generally is composed by several neurons and interneurons that can have between them feed-forward connections and feedback connection that can stop the firing of the neuron. This is really interesting because there are several models showing that pretty basic circuits between parvalbumin neurons and pyramidal neurons and also somatostatin interneurons can produce this kind of behavior. Okay, so basically the parvalbumin neurons is basically giving the timing of activation of the pyramidal neurons, and somatostatin neurons can be this kind of break to this activity.
Okay, perfect. Yeah, so in that regard, there could also be sort of like local networks of, let's say, reverberant activity of inhibitory neurons, right? Like, which is what produces the gamma
yes.
rhythm. Yeah.
Yes, it's very well described for gamma oscillations, which are basically the major activity that you can measure in the brain when you are performing working memory tasks.
Yes.
So yeah, it can be the mechanism.
All right. Okay. Let me see if we have another question. Yes, we have another one from Luana Wozniak. So by increasing the influx of calcium, could there be a potential damage because of cell swelling on the brain based on the sodium channels activity?
That's an important and interesting question. Yes and no. Basically, the idea is the calcium remains high for brief periods of time, but in a regenerative way. Okay, so that's the clue of this effect. Okay, and also we cannot discard the idea that calcium can be the playing a major role in this activity by directly activating channels, but also can be indirectly modulating the activity of the channels present in the neurons by post-translational modifications such as inducing phosphorylation and dephosphorylation of the channels, which can also modulate the activity of the channels. Okay. And the last part, it is well known that several channels can be activated by this increased or constant influx of calcium. Okay, so also this channel TRPM4 has been described in the excitotoxicity. Okay, and several other TRP channels have been involved also in this cell swelling and produced basically by the excitotoxicity.
All right. All right. Thank you so much, Dr. Leiva, for that excellent talk. And I think we have come to the end of today's session. And we are looking forward to see everyone tomorrow. Tomorrow we'll be starting back at 9:15 a.m. And I think that will be a different link sent to everyone who has signed up so that you can join to the sessions tomorrow. So stay tuned to your emails. And thank you so much for attending today. And we'll see everyone tomorrow at 9:15. Thank you so much.